Also, humble request, kindly keep your phones on silent mode so that there is no disturbance during the sessions. Thank you very much.
यहाँ प्रेजेंटेशन yes. नहीं हो रहा फिर तो इसमें मट्टी क्या हो रहा है ये सामने किसको लगा हुआ है ये तो मैंने पहले बोला था इसके और सामने लगा यहाँ फ्रंट में हाँ
A humble request to our dignitaries, to our guests who are outside, to kindly join us back in. We are about to begin our event. Also, a humble request, please stick to your presentation timing, as we have other presentations lined up also, and we would like that everybody gets a fair chance. Thank you so much. to all. Respected Chief Guest, Professor Shiv Serene, Professor N.K. Ganguly, Professor G.P. Talwar, and Professor Hiroshi Ichimura, our esteemed chairpersons, speakers, delegates, senior international colleagues, and our young investigators. It gives us immense pleasure to welcome you for the Series 13th India Probiotic Symposium. The theme of the symposium this year is Science-Based Evidence on the Benefits of Probiotics for Human Health. We are pleased to share that more than 1,200 delegates are participating online and more than approx 300 are in the hall with us here today. We hope you enjoy the presentations during the next 2 days. We will now proceed with the lamp lighting ceremony, which is a typical tradition in India and marks the commencement of any important event. We are honored to have among us Professor Shiv Serene as the chief guest. May I please invite Professor Shiv Serene, Professor Nirmal Kumar Ganguly, Dr. S.M. Shastri, Professor G.P. Talwar, Professor Hiroshi Ichimura, Dr. K. Kitahara, and Mr. Takuya Sugimoto to kindly come forward and light the lamp, please. Let's welcome our dignitaries with a round of applause, please. Thank you so much.
Almost nearing 97.
On the stage for the inaugural session. Yes, sir, please.
Thank you.
Thank you to our esteemed dignitaries for being part and parcel of today's event. It is a delight to have each and every one of you present here with us. Thank you once again. May I now request Dr. Neerja Hajela, General Secretary, Gut Microbiota and Probiotic Science Foundation India, to kindly deliver the welcome address. Dr. Neerja Hajela has done her PhD in biotechnology and has a special interest in probiotics. She is currently the General Secretary of the Gut Microbiota and Probiotic Science Foundation India that aims to establish credibility for the science of intestinal microbiota and probiotica in the country. She has over 17 years of experience and is involved in development, planning, and implementation of scientific programs. She is associated with the Asian Federation of Societies for Lactic Acid Bacteria. She is a member of the Scientific Committee of the Probiotic Association of India and a member of the Governing Council for ILIS India Knowledge Center for Functional Foods, Gut Health, and Immunity. Let's welcome ma'am with a round of applause, please.
Thank you. Thank you, Chandra. Thank you very much. Good afternoon and a very, very warm welcome. Respected chief guest, we don't have him with us, but he has been the man behind this show, Professor Shiv Sareen, Professor Nirmal Kumar Ganguly, and we are honored to have Professor G.P. Talwar. A big round of applause for Professor G.P. Talwar. Professor Hiroshi Ichimura, our esteemed chairpersons, international national speakers, panel moderators, panel members, delegates, senior international colleagues from Japan, and our very own young investigators. It is an honor and privilege to deliver the welcome address. At the outset, we would like to thank Dr. Shiv Sareen, the director of the Institute of Liver and Biliary Sciences, for giving his consent to co-host this symposium. I remember when Professor Ganguly and I went to meet him, he was delighted and so very enthusiastic and immediately agreed to organize this scientific feast. We are privileged and honored. We would also like to thank Dr. S.M. Shastri, Additional Professor, Department of Hepatology, and his team for their tremendous support in organizing this event. Dr. Shastri, despite his very busy schedule and his endoscopies and so many other things, has been with us at all times, and without him, this symposium would not have been possible. The symposium is an initiative of the Gut Microbiota and Probiotic Science Foundation India and is being co-hosted, as you can all see, by the Institute of Liver and Biliary Sciences. The foundation was established in 2011 under the presidency of Professor Nirmal Kumar Ganguly as a society under the Societies Registration Act 21 of 1860. Professor Ganguly, a visionary and outstanding scientist, and has been the brain in establishing the foundation and has held the torch and shown us the path. We are ever so grateful to him for his wisdom, guidance, and unflinching support. A big round of applause for him, please. Our foundation members, Dr. B. Sesikaran, Professor G.B. Nair, Professor A.K. Srivastav, Professor Ajay Bhalla, Professor K. In the pillars and the guiding force who have helped us reach thus far. For all our members, governing body members who are here in the audience, could you just stand up or just raise your hand so that we can have a big round of applause for all of you? We would also like to thank our co-opted members for constantly encouraging us and funding the foundation so that the science can be widely recognized. A big round of applause for our co-opted members, Japanese members, please. The primary objective of this foundation is to share the latest developments in the area of gut microbiota and probiotics, a science that is evolving at a rapid pace. 2023 has been yet another big year for microbiome and probiotic research. Not only is funding at an all-time high, but the shift from 16S to whole genome sequencing means that we are able to understand the role that these microbes, and we know that we have 100 trillion microbes, play in health and disease much, much more effectively. There are whispers of a skinny microbiome fixing the gut, the amniotic and the placental microbiome, and science is advancing at a pace that we had never, ever imagined. As a result of this knowledge and understanding, interventions like probiotics, prebiotics, and symbiotics, which favorably modify the intestinal microbiota, have taken center stage. Their functional and mechanistic aspects are also being better understood. Several meta-analyses and systematic reviews suggest that such interventions could be a promising method of restoring a healthy gut microbiota, which may then help in better management of gastrointestinal, extraintestinal disorders like metabolic syndrome, inflammatory bowel disorders, cancer, and respiratory disorders. The book is also now opening on the gut-brain axis, and these microbes may be linked to brain function and behavior. Two large landmark studies from India have shown that the benefit of probiotics in reducing incidence of diarrhea and neonatal sepsis, both leading causes of mortality in infants and children. People are embracing the microbiome diet, and fecal microbiota transplants are transplanting the gut microbes from a healthy donor to a diseased recipient, maybe in the next frontier in science for treatment of disease. So as the foundation forges ahead to advance the scientific knowledge of the intestinal microbiota and probiotics, it gives us immense pleasure to welcome you for the Series 13th India Probiotic Symposium on science-based evidence on the benefits of probiotics for human health. The presentations by international and national experts will throw light on the role of the gut microbes on the aging immune system in neurological disorders, psychological stress, and anxiety. It will also highlight changes in the gut microbiota composition in diarrheal patients and healthy controls from a slum dwelling in India, and microbiota changes in responders and non-responders, which can help in clinical decision-making in oncology. The speakers will also deliberate on the possible use of probiotics for HIV-infected children, for the athletic population, and as vaccine immunity boosters in immunization. They will discuss how such interventions may help in reducing the risk of non-communicable diseases and post-operative infectious complications. There will also be a talk on the use of these interventions in antimicrobial resistance, which is a serious global concern today. Therefore, there will be huge learning in these 2 days. The symposium has been made all the more interesting because of the original research being conducted in India, which has been displayed as posters by our very own young scientists. These young scientists were selected after a rigorous review of their work by the foundation members and an international jury with Professor Hiroshi Ichimura, Professor, Faculty of Medicine, Institute of Medical, Pharmaceutical and Health Sciences, and Emeritus Scientist, Kanazawa University, and Mr. Takuya Sugimoto, a senior researcher at the Analytical Science Department, Yakult Central Institute, Japan. The top 3 scientists will present their work as oral presentations. The posters have been displayed just outside the venue. We encourage and urge every one of you to view the posters for the kind of work that is being undertaken in the country by our own young minds. The foundation has also been bringing out the proceedings of the past symposia in the form of a book with an ISBN number. The books have been published by Elsevier, Wiley, and this year by JP Medical Publishers, who have done an excellent job. This book, Gut Microbiome, Probiotics and Gut Health, The Evidence Gets Stronger, will be launched today by our distinguished dignitaries and distributed to healthcare professionals, students, and libraries in the country. And they will serve as a repository of information. We would like to thank the contributors and the editors for their immense effort and time. We also request you to visit our mobile-friendly foundation website, that is www.gutfoundation.org.in, which is a valuable source of information for students, for scientists, researchers in the area. The presentations of the previous symposia have been archived on the website of this foundation. On behalf of Professor N.K. Ganguly, all members of the Foundation and the Institute of Liver and Biliary Sciences, we once again thank each one of you individually and all of you collectively for having taken the time to attend this symposium. We hope you will enjoy the presentations, discussions, deliberations, and the new science that will ensue over the next 2 days. Thank you so much.
Thank you very much, ma'am, for the wonderful welcome address. May I now request Professor Nirmal Kumar Ganguly, the President of Gut Microbiota and Probiotic Science Foundation India, to kindly deliver the opening remarks. Professor N.K. Ganguly needs no introduction. He is a former Director General.
No introduction will. Thank you. Thank you. We will save time.
Okay.
So thank you, everyone. Thank you, everyone who have come here. You have made me very, very happy, particularly all the young persons who want to absorb knowledge and have come here in great numbers. We are also very thankful to the poster presentees, the 21 of them who are here today to interact with all of us. And their posters are so good that previously we had thought that we will do it with 4 of them, but later on we thought that we need to interact with all of them because all the posters were very good. Whether it won a prize or it didn't win a prize, every one of them was very, very good. So many of the youngsters, the seats in front are vacant, so you could move forward. You will be able to see the slides better and perhaps hear better. So I will start with introducing Dr. Talwar's work because he will not be speaking. So since I'm here, and it gives me great pleasure to introduce to you one of the original worker who worked in this field. Dr. Talwar, in his own wisdom, thought that his special interest was in the reproductive tract because he invented the first contraceptive vaccine, that is a beta-HCG vaccine, which at that time was a great idea that if you give one shot, the ladies will not have to come again and again. And you go with your baby to the well baby clinic, get a shot, then you don't have to ask your mother-in-law or your husband that whether you need a pill or you don't need a pill. So it was a great idea. And the vaccine is a great vaccine. It is not only working in the contraceptive field, it is working in other fields. So this was because this was his main interest. He started working on the microbial flora, which populate the genital tract. As you know that the health of the genital tract is maintained by the microbial flora because the pH there has to be acid pH. And if the pH becomes alkaline, then the environment becomes bad and all kinds of infections, all kinds of inflammation sets in. So before he worked on that, this dogma was not known. People thought that these are populated by the gut microbes only, and they are the population which is there. So there is nothing new in that. But he identified that there are special cellulophores or receptors with which there are unique population over there. And this population will be studied. So he identified those populations with painstaking work and then created a probiotic mix for the genitourinary health. So vaginosis, vaginitis, etc. Because 60% of the Abhay and Rani Bang, who work in the Gadchiroli, first published a paper that 60% of the women in India, they have white discharge. So they have some kind of vaginitis or vaginosis. And that affects their reproductive health, that affects their children, that causes also abortion or low birth weight babies or many other issues, and also the inflammation of the reproductive tract. So he created a product and he took it to the company. Company produced it. It went to the clinical trial. So he is a person who is ultimate translational guy. His discoveries ultimately go to company. And we were very grateful that he really did it the first time globally. After that, there was an Italian scientist known as Carlos Miotti. He developed another product, the same principle. He named it as a Floretia. And he also developed a gloves which turns color if the pH becomes high. So this is something which is Dr. Talwar's work on the probiotics of the genital tract. And we are very grateful for your interest. He still works. By the way, he has a laboratory just besides his house. His children said that you could use your money. We don't need your money. You could open a laboratory. And he does active research. He still publishes. How he does it, I don't ask me. I do not know. But he does it. So hats off to Dr. Talwar. Since you were not speaking, I thought I give some of your work to the younger generation who should get inspired by your work. As you know that we were working in this area for many years, and through this foundation we wanted to promote this science. This science is now coming to a great clinical application. Yesterday we had a workshop. First time we never did any workshop. We did many activities, but not workshop. But because of Dr. Shastri and Dr. Sareen, we did a workshop on the FMT yesterday. And it was revealing where the science has now moved to the clinics, to the patients. And it is not that you give some pills or you deliver it through food. You really work different kinds of clinical situations with the fecal transplant or perfusion. In this hall only, we had brought in a Japanese surgeon who was transfusing the probiotic mixtures into patients to get a better result, who underwent colon cancer surgeries, and the effects were great. So the science is now really moving to clinics. In the dairy science, it has moved to application much before we really did it over here. But in the medical science, but it is now moving very fast to the clinics. The science has also moved at a breakneck pace. The maximum number of papers are published in this, and we are now understanding much more about how these guys actually control every part of our life, whether it is obesity, whether it is our fat content, whether how our brain works, how our liver works. You just name it, and these guys control it. So these were helped by three different streams of the science. One was that the metagenomic techniques, with which we could get the entire community and flora and the relationship with each other. This came because of the high throughput sequencing, which was available. And the other thing came from the multiomics, because these guys control your metabolic pathways. These guys secrete a lot of things, exosomes, proteins, and others. And these also ultimately control many of our functions and actions, and they build up the immune system. We will be having a panel today to understand a little more about that. But they build an immune system, whether it is beneficial immune system, because gut immune system is one of the strongest immune system. Whether it is corona infection, whether it is HIV infection, they destroy the gut immune system first to make the people vulnerable. So these guys, they build up the gut immune system beneficially. But then they also reduce inflammation. They also can move, if they are not there, the gut immune system can move towards autoimmune reactions. It could move towards allergy, ectopy, and other things. So if they are not there, it becomes very difficult to survive and live. And the earlier as we go old, the diversity becomes less and less. The diversity is the hallmark of the our healthy gut. And there, the earliest you get it, many people were thinking that when you become old, when you become needy, then you take probiotic. Earliest you are given probiotic, Dr. Mittal and Kyal Heidi will perhaps agree with me. They are then recognized by these immune cells and they start behaving, they get trained. But if they get it at a later part of their life, they look at our immune systems look at these bugs as foreigners, and at that time some of them who has some problem in the regulation of the immune system, they could be skewed others. So this science which has moved now is so rich that I think that this could become now the whole and complete career for some people in the medical field. In the Dr. Prajapati and Dr. Srivastav, etc., who are coming from dairy field know this dairy field. But then we have lot of people from the nutrition field over here. These are also providing essential nutrients to us, their absorption, facilitating gut environment. So if we really look at this becomes a very, very important part of our life, an important part of science, which will make us and give us our bread. So all you youngsters who are sitting in back seats, actually they look so full, please come forward. You will find a very good career into this area, very rewarding career in this area. You need not have the instruments, they are available. Just think about it and you will see how it works. One last word I will like to say, that they differ in the different population. If you go to Japan, if you go to Denmark, if you come to India, you will find that different communities and different populations are over there. And sometimes they could unravel that why the Japanese are living the longest in the world, why Italians have lesser heart conditions. So many things we could understand by the behavior of these guys. So these guys are friends and these guys are enemies. It is up to you whether you want to make it a friend or you want to keep it as an enemy. So this is what we will learn today. As Nirja has said, we have a tremendous feast awaiting us today. I invite you to have this. The lunch was not the main feast. This is a feast. So enjoy it completely and fully and remain in the science. Last of all, I will like to thank Yakult Honsha Company, which for last 11 years, without any string attached, has been providing us fund to be functional. And I'm so grateful that they have not even withdrawn this particular donation at any given time. And this is absolutely without strings. So thank you very much, Mr. Amano, and all of the company who has supported us all the time. Thank you.
Thank you, sir, for your words of wisdom. It was truly a delight to hear your insights. Can we have a round of applause for sir here? That was really quite commendable indeed. I would now be requesting Professor Shiv Serene to kindly deliver the inaugural address. Professor Shiv Kumar Serene is the Senior Professor, Hepatology, Chancellor and Director of Institute of Liver and Biliary Sciences, New Delhi. He was instrumental in setting up the institute under the auspices of the Government of Delhi. He is the Director, WHO Collaborative Center on Chronic Liver Diseases and Viral Hepatitis at ILBS and Preston, National Academy of Medical Sciences. He serves as adjunct faculty molecular medicine at JNU. Due to his deep interest and contributions to translational science, he is credited with several new treatment protocols for liver diseases. He has more than 760 publications to his credit, edited 13 books on liver diseases, and contributed 90 chapters in various medical textbooks. Let's hear him out now.
Sir. I'll go down to see these slides better. Please.
Greetings from the Institute of Liver and Biliary Sciences. I'm indeed very grateful to respected Professor Ganguly for having organized this meeting at ILBS. It's a privilege for us that we can host this meeting on probiotics. I am associated with this organization for a long time, and I know how excellent work the organization has done under the leadership of Professor Ganguly. And this National Symposium is a testimony to the growing eminence, relevance, and reach of this National Symposium. Dr. Nirja has done excellent work for us and for the organization. I would humbly say that ILBS has done some work in this direction, not exactly as probiotics, but in the field of gut microbiome, especially in relation to alcoholic liver disease. ILBS was the first in the world to start fecal microbial transplant in 2013 using a nasoduodenal tube and giving fresh fecal slurry from relatives, healthy relatives of alcoholic liver disease patients. And we have done 4 trials on that. First was a pilot study of 8 patients, then a study on comparing this with pentoxifylline. The third study was in steroid eligible patients to find out whether the fecal microbiota transplant can be an alternative to steroids, and it was. In fact, there was fewer infections and better outcomes, and the overall behavior and health of the patients was better. And the last study, which is undergoing now currently, is in patients who are ineligible for steroids as a randomized trial. With that, we also do FMT in many other groups of patients, especially with primary sclerosing cholangitis, or sometimes even in patients with decompensated advanced NAFLD cirrhosis. We also have very good labs where we can do not only the 16S, but metagenome and metaproteome of bacteria, and can differentiate between bacterial and host proteins, metabolites, and extracellular vesicles. The current symposia, with so many outstanding, distinguished speakers from all over the world, will certainly result into I would love to be with all of you, but due to some prior commitment, I have to miss this very important international and national symposia. My colleague, the Vice Chancellor, Professor Deepak Tempe, and the whole team from hepatology and basic sciences would join. My greetings and salutations to the whole organizing team, and welcome again to the delegates from overseas, from India, and to all the faculty members. And I wish a very fruitful and a very educative and continuing experience. Thank you all for choosing ILBS and making us part of this very important event. Thank you all.
Move ahead to the next segment of our event. We now have the book release gut microbiome, probiotics, and good health. The evidence gets stronger. May I now please request our dignitaries herewith to kindly step forward for the same, starting with Professor Nirmal Kumar Ganguly, Professor G.P. Talwar.
दीपक टेमरे हैं तो दीपक
Yes, sir.
टेमरे हैं
Mr. Deepak Temre as well, Professor Hiroshi Ichimura, Professor K.A. Lehri, ma'am, to please join us here as well, Dr. S.M. Shastri, Dr. Kae Kitahara, and last but not the least, Mr. Takuya Sugimoto, to kindly step forward for the book release now. Everybody, can we please have a round of applause for our dignitaries here on stage? Go one by one. Ma'am, please. Everybody, can we please have a round of applause? Louder. I would request our dignitaries to kindly get ready for a photo op here. Thank you once again to all of you. We would be taking a short break. I would request everybody to gather for group photograph followed by tea break. And please be here back at 15:30 p.m. sharp so that we can commence the next session on time. Thank you so much. Welcome once again to our honorable guest and dignitaries. Post our break time, we are back to begin our next session, Scientific Session 1. I would be requesting the humble presence of our chairperson for this session now. Professor Rama Chaudhary and Professor V. Samuel Raj were presiding over this session. Professor Rama Chaudhary has done her MBBS from Lady Handring Medical College and MD in microbiology from AIIMS, New Delhi. She was the former head of microbiology at All India Institute of Medical Sciences, New Delhi. She has an experience over 35 years in the field of academics and medical research, and she has over 200 research papers published in journals of both national and international repute. She has been associated with the research advisory and project review committees of national funding sectors such as DBT and ICMR. Currently Senior Director and Professor Microbiology and Dean Research, NIMS University, Jaipur. A hearty welcome to you, ma'am. Can we have a round of applause for her, please? Also, our other chairperson for this session herewith, Professor V. Samuel Raj, is working as the Professor of Microbiology and Biotechnology and Director of the Center for Drug Design Discovery and Development, C4D, at SRM University, Haryana. He joined the SRM University, Haryana with 10 years of R&D experience from two major pharma giants, Ranbaxy and Daiichi, and 15 years of international and national research experience. He has a special interest in neglected diseases. He has won many awards and fellowships in the USA, Japan, Switzerland, UK, Taiwan, and India, including the prestigious Tokyo Biochemical Research Foundation Award and Commonwealth Fellowship. Can we have a round of applause for Sir here as well? I would request our chairpersons to kindly take the lead here and take the session forward. Thank you.
So good afternoon, everyone. We will start the first session of today's conference, the 13th India Probiotic Symposium. And the first speaker is Professor Hiroshi Ichimura. Dr. Hiroshi Ichimura is the Professor of Faculty of Medicine, Institute of Medical, Pharmaceutical and Health Sciences, Kanazawa University. He is also an Emeritus Professor of Kanazawa University. After obtaining his MD and PhD in Japan, he did his postdoctoral training at Baylor College of Medicine, Texas in 1985 to 1987, and at the University of California, San Francisco in 1991-92. He became the Professor of Department of Viral Infection and International Health, Graduate School of Medical Sciences, Kanazawa University in 1999. He's also an honorary professor at Hanoi Medical University and Hai Phong Medical University, Vietnam. He received Medal for People's Health from the Ministry of Health, Vietnam in 2016. He serves as a director and counselor at the Japanese Society for Preventive Medicine, the Society of Japanese Virologists, and the Japanese Association for Infectious Diseases. He has mainly studied viral infections such as HIV/AIDS, human papillomavirus, and hepatitis in Kenya and Vietnam. Dr. Ichimura will speak on effects of short-term probiotic ingestion on immune profiles, microbial translocation, and gut microbiota among HIV-infected Vietnamese children. Welcome, Dr. Ichimura. You please proceed.
Good afternoon, Professor Ganguly, ladies and gentlemen. This is Professor Hiroshi Ichimura from Kanazawa University, Japan. Thank you very much for your kind introduction, Professor Chaudhary. So I'm very honored to have an opportunity to present our data in this symposium. So today, I'd like to talk I will talk about the effects of, it's not, I cannot see it. Is it possible? No, sir. No. No. Okay. So I will talk about the effect of short-term probiotic ingestion on immune profiles, microbial translocation, and gut microbiota among children with HIV infection. Human immunodeficiency virus type 1 HIV mainly infects CD4 plus cells, and the gut-associated lymphoid tissues, GART, is the largest HIV replication site and reservoir. In HIV-infected individuals, CD4 plus T cells in the gut are reduced and the immune control is lost, resulting in dysbiosis, disruption of the intestinal barrier, and chronic immune activation due to bacterial translocation. As you know, the effective antiviral drugs have been developed. However, even if anti-HIV therapy, ART, is initiated, gut immunity is not fully restored and dysbiosis persists. So probiotics and diet are being tried to restore a healthy gut. So well-known effects of probiotics include enhancement of epithelial barrier, increased adhesion to intestinal mucosa, inhibition of pathogen adhesion, competitive exclusion of pathogenic microorganisms, production of anti-microorganism substances, modulation of the immune system. A probiotic strain, Lactobacillus casei strain Shirota, LCS, has been used to make the fermented dairy product for over 80 years, and in Japan and many other countries, including India. The Yakulto, a formula milk product containing LCS, is known to have the function to modulate the bacterial microbiome in gut, to enhance the barrier function of gut membrane, and to intensify the innate immunity in gut. Thus, LCS injection is expected to improve the pathogenesis of HIV infection related with gut and the efficacy of ART. In collaboration with Yakulto Central Institute, so we investigated the effects of short-term probiotic LCS injection in HIV-infected children who had been followed up in Hanoi National Children's Hospital in Vietnam. So I would like to introduce the result in this talk. So the aim of this study was to elucidate the effect of short-term probiotic ingestion on the immune profile of microbial translocation and gut microbiota in children with HIV infection. It was estimated that there were 3,900 HIV-infected children alive in Vietnam at the end of 2011, of whom more than 500 people, children, were being followed at the hospital. So in this study, 60 HIV-infected children were included. Of these, 29 were treated with ART, with median ART duration of 36.8 months. 31 were untreated. And other control, 20 uninfected healthy children were included. And each group were designated as ART plus, HIV plus, and HIV minus. So these children ingested the LCS once daily for 8 weeks. So, blood and stool samples were collected at baseline for 8 weeks after initiation of LCS and 4 weeks after cessation of LCS injection. So Yakult drink was delivered to each study site by Yakult Vietnam once a week for a total of 8 times. In the HIV plus group, the family of the parents of each subject visited the hospital every week to receive Yakult drink for his or her child. The children in the ART plus group and HIV minus group stayed at the above this center and another center in Hanoi. And therefore, the children in the same kind of group basically had almost the same kind of diet. Whereas the HIV plus group stayed at home in each home. Therefore, we could not control their diet well. So whole blood samples are used to assess the activation state of CD4 and CD8 cells, and CD4 cells and the CD4 subset proportions using cell staining method, targeting specific surface markers. So plasma samples were used to examine HIV viral load and to assess bacterial translocation via IF-scan, which measured bacterial ribosomal RNA molecules and ribosomal DNA, targeting 14 enterobacteria. Stool samples were used to analyze the gut microbiota by RT-qPCR. So in this study, we analyzed CD4 subsets and to assess the CD4 proportion and the activation of CD4 and CD8 positive cells using cell staining methods according to the method of the Nature Review Immunology. CD38 positive and HLA-DR positive cells were designated as activated CD8 and CD4 cells, respectively. And the CD4 subsets were classified as Th1, Th2, Th17 using CCR6 and CXCR3 as indicators, and regulatory T cells, Treg, using CD25 as an indicator. So let me move on to the result. So first, let me introduce the effects of short-term LCA injection on physical and clinical parameters in children with HIV infection. So after LCS injection, height, body weight, total cholesterol, fasting blood sugars, all are increased in HIV plus group. And interestingly, AST and ALT were decreased, significantly decreased after LCS injection in HIV plus and the ALT plus groups. So this is a summary of the change in physical and clinical parameters associated with LCS injection. So after LCS injection, no serious adverse events occurred. Total cholesterol, blood sugar, height, and weight increased significantly in the HIV plus group, suggesting that LCS supplemented fermented milk benefits improved nutrition and growth of the HIV plus group. So ALT and AST significantly decreased in HIV-infected children, suggesting that LCS may have protective effects on the liver. Next, let me introduce the effects of LCS injection on immunological and viral parameters in the children. So as mentioned above, HIV-infected individuals show a decrease in CD4 plus T cells, but impact of HIV infection and ART on CD4 subset and cell activation in children with HIV were not well understood. So we first analyzed them as the baseline characteristics of the study participant. So this figure is a summary. HIV infection induced caused a rapid decrease in Treg cells and early activation of CD8 plus cells and monocytes. So ART induced rapid recovery and early normalization of the CD8 plus cells, cell activation, but had little effect on monocyte activation. This slide shows the change of CD4 plus T cells and their subset after LCS injection. So CD4 plus T cells and Th2 cell T cell count increased in the HIV plus and the ART plus group, and Th17 increased in all the groups, whereas Treg cells decreased in ART plus and HIV minus group. So CD4, CD8 counts. So this is the marker of the to predict the beta kind of therapeutic control, and the beta immune status just increased in all the groups. And interestingly, HIV plus group, the plasma HIV viral load slightly, but statistically significantly decreased after 8 weeks LCS injection. After LCS injection, CD8 cell in HIV plus group, CD8 cell activation has significantly decreased, but after cessation of LCS, the CD8 cell was abnormally activated again. This rebound also was observed in CD4 plus cells. However, so LCS injection did not affect any monocyte activation. So this is the summary. So after LCS injection, CD4 plus T cells, especially Th17, increased in all the groups, and Th2 increased in the HIV and ART plus groups. Treg cells decreased in the HIV plus, ART plus, and HIV minus group. CD4, CD8 increased in the ART plus and HIV minus group. So plasma viral load slightly but significantly decreased in the HIV plus group. So abnormally activated CD8 cells normalized in the HIV plus group. So that's the LCS injection induced a better immune status in children with HIV infection. We analyzed the ribosomal RNA molecules in whole blood to detect viable bacteria by RT-qPCR. and ribosomal DNA levels in plasma to detect both viable and dead bacteria by qPCR. So in the RT-qPCR analysis of blood, so we analyzed. In the RT-qPCR analysis of blood bacteria, these are red ones. So Pseudomonas was detected only from one subject in the HIV plus group after LCS injection for 8 weeks, which indicated that bacteremia is very rare, even in children with HIV infection. So then we analyzed the bacterial ribosomal DNA in plasma by qPCR. So ribosomal DNA from Staphylococcus and Streptococcus and the Pseudomonas was detected in the HIV plus group. And Staphylococcus 16 ribosomal DNA detection was 0% in the ART group compared to which one is much significantly lower in the HIV plus group, 22.6%. So this one suggests that the effect of ART to suppress bacterial translocation in HIV-infected children. So this is the result of qPCR analysis of plasma before and after LCS injection. So as you can see, so many Enterobacteriaceae, Streptococcus, Enterococcus, and Staphylococcus, and Pseudomonas, ribosomal DNA, ribosomal RNA was detected, ribosomal DNA was detected in plasma in all the groups. However, no significant change was observed in the detection frequencies between before and after the injection. Next, so let me, then we also investigated the effect of short-term probiotic injection on gut microbiota in children with HIV infection. This is the structure of gut microbiota at baseline and after LCS injection. As you can see, principal component analysis PCA revealed that the structure of the gut microbiota in each group slightly changed, but did not change significantly from baseline. So this one is the change in gut microbiota after LCS injection. So lactobacillus species, after LCS injection, lactobacillus species and bifidobacterium significantly increased in the HIV plus and the HIV plus and the ART plus groups. In the ART group, the remarkable decrease in CPAP ranges was observed. This slide shows the association of intestinal microbiota with age, duration of ART, and immune markers at baseline before LCS injection. So in the healthy children aged 2 to 8 years, subdominant bacteria such as Streptococcus, Enterococcus, and Enterobacteriaceae were significantly associated with age, but dominant intestinal bacteria were not. So in the HIV plus group, these associations disappeared, and the number of Staphylococcus was negatively associated with age. So in the HIV positive groups, their age nearly equaled the HIV infection period because their HIV-infected children just acquire the virus vertically from their mothers. So the number of Clostridium coccoides was positively associated with those of CD4 plus T cells and their subset. In the ART groups, Staphylococcus and C. perfringens correlated with the recovery of CD4 plus T cells and their subset, and normalization of activated CD8 plus T cells. So we do remember this one, C. perfringens strongly correlated with ART durations. So this is a change in gut microbiota after LCS injection in association with ART duration and immune markers. So in the ART groups, increase in lactobacillus species increased after LCS injection. Increase in lactobacillus species and bifidobacterium were associated with Th17 recovery. And changes in the C. coccoides group were associated with increase of Th1 and Treg. Decreases in C. perfringens was strongly associated with the duration of ART. This figure shows the association between between the number of C. perfringens and the ART duration in the ART plus group. You can see in the ART plus group, C. perfringens was significantly decreased after LCS injection from purple dot to green dot. And the rate of decrease in C. perfringens was significantly higher in children with longer duration of ART. This is a summary of the effect of LCS on gut microbiota. So let me discuss one by one. So we found after short-term LCS injection, lactobacillus species and bifidobacterium slightly increased in the ART plus group, and the increase was positively correlated with Th17 recovery. So these findings could be partially explained by the previous reports that lactobacillus species and bifidobacterium produce lactic acid and acetic acid in the gut, which contribute to intestinal barrier function, regulation, and activation of intestinal immunity, including Th17. And that LCS is also directly involved in regulating Th17 differentiation. Second, so in the ART plus group, changes in C. coccoides group after LCS injection positively correlated with Th1 and Treg recovery. So this finding is also inconsistent with the previous report that C. coccoides group produces short-chain fatty acid, SCFA, and plays a central role in the regulation of gut inflammation. The third one, we found that C. perfringens, possible harmful bacteria, significantly decreased after LCS injection. So the decrease rate was particularly high in children with longer ART duration. So considering the SCFA is known to inhibit the growth of C. perfringens, so we would think that the metabolic products of lactobacillus species, bifidobacterium, and C. coccoides group may have inhibited the growth of C. perfringens. In conclusion, our findings suggest that short-term LCS injection can be a safe, supportive approach with immunological, nutritional, bacteriological, virological benefits in HIV-infected children, especially those on ART. So these are our collaborators. Thank you very much for your attention.
Thank you, Professor Hiroshi,
Yeah.
for elucidating nicely the action of Lactobacillus casei shirota strain, which was isolated in Tokyo, Japan, and which is actually the strain present in Yakult drinks. So this particular preparation can have effect on the gut microbiota as well as on the immunity, which was shown by the studies done by RT-PCR on the gut microbiota, as well as by doing the, I think, flow cytometry study by seeing the different cell population in the immune cells. So I think it's, though the number is not very high, but it's a significant data. In fact, one of the study at AIMS, a PhD student has also shown that in HIV positive patients, the translocation of microbes can occur from the vagina, and especially in the females, and also in males through the gut, and can lead to a lot of effects on the gut immunity as well as on the microbiota. So I think it's a significant result showing the effect on different T cell population. And if there are any questions, if time permits, we can take one or two questions. Is there any query or questions from the audience?
Yeah, good study. Any comment on why Treg was lowered? You have good TH17 during treatment, but I would have expected the Treg population to be equally increased after treatment, but there is a decrease of Treg. Why so? Okay.
He is saying that in your study, the TH17 cells were increased, but the T regulatory cells were not showing increase. So we were expecting the T regulatory cells also should be influenced and should be increased. So what could be the reason for that?
Actually, honestly speaking, the kind of classification of TH17 or the CD4 subset is a bit behind. Currently, we need a little bit more surface markers are needed. So that part may be the reason, could be the reason for the difference.
Yeah.
Yep.
So I think the question is very relevant.
Actually, what was happening, Professor Chaudhary, that TH17, there was a recovery because it is one of the major mucosal inflammatory pathogenic molecule. But Treg increase causes immunosuppression. So decreasing Treg actually restores immune functions. And the short chain fatty acids, what he said was one of the major reason for this activity which was observed. So actually Treg in the recovery of the immune system, because Tregs are the suppressor cells. So Tregs, if they decrease, there is a recovery from the immune system. Tregs are the suppressor molecules. So that is how it worked. But one of the best thing which happened was this we see after FMT, AST/ALT recovery is one of the very strong clinical markers which you saw. But one of the major challenge will be that these were pediatric population.
Yes.
I don't know whether this could be replicated in adult population or not.
Other populations. Yeah, so I think in adults, there have been several reports but in pediatrics field, so there are less kind of the studies available. So that is the issue. And also I used to work as a pediatrician. So that is one of the reasons to focus on the children.
Thank you. Yes. Please take the mic and
Thank you for your thought-provoking presentation. I have a safety-related question. When you're administering probiotics to AIDS patients, should there be a CD4 threshold below which you should not maybe administer probiotics because of safety concerns?
So I didn't tell you, I didn't tell the AIDS patient, HIV infected children. So so I think the all the HIV infected children have the CD4 count with more than 200 per microliter. So of course we discussed with the Vietnamese pediatricians, and they themselves recruited the appropriate kind of candidates for this study. So I think it's not really somehow immune compromised, but not AIDS conditions.
So what he's saying is that they were not clinically having AIDS, though they were having low CD4 counts, which was like 200. But yes, yes. So the question is relevant. Of course, in severely immunocompromised patients, some of the strains may become cause septicemia. There are some reports, and so one has to be careful. But yes, definitely, this strain seems to be safer and very well studied for a long time.
Yes.
And so I think that's the reason, at least in these patients, the results were positive. Thank you.
Thank you.
So we'll go to the next presentation.
That was quite an impressive presentation indeed. Thank you so much, sir. I would request you to humbly come back onto the stage. We would like to seize this opportunity to felicitate you. So, sir, can we have you here back? And I would request Professor Rama Chaudhary, ma'am, to kindly do the honors of presenting the token of appreciation and memento to our speaker here. Professor Hiroshi Ichimura, sir, kindly please accept this token of appreciation from our end. It is a delight to have you here with us. Everybody, can we please have a round of applause? Thank you. Thank you so much.
So we will go to the next lecture in this series. This will be impact of athlete lifestyle on immunity and potential benefits of probiotics. This will be given by Professor Michael Gleason. He is Emeritus Professor, Exercise Biochemistry, Loughborough University, Loughborough, UK. Professor Michael Gleason is an Emeritus Professor of Exercise Biochemistry at Loughborough University. He retired in 2016 after 40 years of research and teaching mostly related to diet, metabolism, health, and performance of athletes. He has a particular interest in the effects of diet and exercise on the function of the immune system, and has provided advice on minimizing risk of infection and nutritional strategies to maintain immune function and optimize performance to numerous sports clubs and organizations. He has published several books on exercise biochemistry, sports nutrition, exercise immunology, and healthy lifestyle. As well as he has over 200 research papers to his credit. and has contributed to recent IOC, ISEI, ISSN, and UEFA consensus reviews. So we are very fortunate to have Dr. Michael Gleason. He will be giving his online presentation. And I think the topic is really interesting for everyone, because ultimately it is diet and exercise which is going to give you healthy microbiota, even if you don't do anything else. So we welcome Dr. Michael. Dr. Michael, for your presentation.
Hello, I'll just share my screen if I may. I can find it. There we go. Slideshow. Well, thank you very much indeed for that introduction there. Thank you. And greetings from the UK, where it's sort of mid-morning right now. And many thanks for the invitation that you've given to me to speak today. As the chair said, some of you may know me from the books I've written to do with exercise biochemistry, immunology, and sport nutrition. I've also done some studies on probiotic interventions and infection risk in athletes. So my talk today is about the impact of athlete lifestyle on immunity and potential benefits of probiotics. So let's begin by just thinking actually what athletes want to be able to do. Well, firstly, of course, they want to be able to perform well in competitions. They want to train well and effectively and maintain a consistent level of performance throughout a season. They also need to be able to recover quickly and avoid burnout. And importantly, they need to remain healthy and avoid injury, because illness and injury, quite simply, are the two most common causes of missing training sessions or missing competitions. In fact, about a third of missed training sessions are due to illness, according to a survey by UK Sport. And around half of these are respiratory infections, and the other main cause of infection that athletes suffer from is gastrointestinal infections. Now, if we think about what the athlete does, they exercise and they need energy for that. And so they eat more than most of us. So they have a high energy intake and protein intake, plenty of carbohydrate to fuel their performance and allow adaptation and recovery. That, of course, means that their gut microbiota actually receive a very plentiful supply of indigestible fermentable carbohydrates, proteins, polyphenols, and all the other microbe accessible nutrients that help to support the diversity, stability, and abundance of the gut microbiota. So we'd expect athletes to have a fairly healthy microbiota. And in fact, exercise is known to support this, because exercise itself independently increases microbial diversity and the abundance of metabolic pathways, particularly those associated with the production of short chain fatty acids. And we can see from data on the microbiota of athletes compared with either low BMI or high BMI sedentary controls, that athletes have greater phylogenetic diversity in their microbiota. And they also have a greater relative abundance of microbial metabolic pathways, again, particularly those associated with microbial derived short chain fatty acids, which can also serve as a fuel for the muscles for the exercising athlete. And these differences between athletes and sedentary controls appear to be related mostly to their higher fitness and also the amount of protein they eat in their diet. And of course, these changes, these improvements in diversity and abundance of metabolic pathways mean they have a nice, resilient gut microbiota, which we would expect to enhance overall health. However, despite this apparently healthy gut microbiota, we find that athletes who are really undertaking very high training loads, particularly during intensified periods of training and during competition, that their risk of upper respiratory tract infections increases several fold. And this has been shown in hundreds of studies, and these have been summarized in these review articles that are cited here. And it's not just exercise, although originally it was thought it was just exercise, and the stress of that, the increase in stress hormones, anti-inflammatory cytokines, etc., causing a depression or lowering of immunity. But we now appreciate that it's actually a combination of a wide variety of factors, and between some of these factors there are reinforcing interactions. So for example, exercise itself, when it's prolonged and hard, can temporarily lower immunity. But when athletes are training hard, their sleep quality is also impaired, and this in itself can also lower immunity. Similarly, sleep can also be impaired by what we might call psychological stress. So that's another impact, that one reinforces the other, as well as having their independent effects on lowering immunity in athletes. And other factors that can be involved are exercising in environmental extremes, which makes the exercise more stressful, traveling long distances for foreign competitions, and also certain nutritional deficits or deficiencies, particularly in micronutrients like iron, for example, or vitamin D. And in female athletes, perhaps also a concern about, are they getting enough energy intake? And low energy intake may also lower immunity as well. So when it comes to picking up respiratory infections, particularly of the upper respiratory tract, we're talking really about an accumulation or a combination of stresses for the athlete associated with their lifestyle, which can depress immune function and therefore make them more susceptible to infection. And of course, the other main factor in infection risk is the degree of exposure to pathogens in the environment. And there are some reasons why this can be increased in athletes compared to the sedentary population, again, according to their lifestyle behaviors. And also we should mention also that, as well as getting sore throats and things like that associated with upper respiratory tract infection, this can also be caused in athletes, those same sorts of symptoms, via an allergy or inflammation of the airways when they exercise hard in cold, dry air, or even polluted air. If we look at the actual incidence of infection amongst athletes, and this is data taken from games like the Olympics lasting 2 to 3 weeks or so, we see that on average about 7% of the athletes experience at least one episode of illness during that 2 to 3 week period. And about 50% of those are respiratory infections. In fact, illness incidence actually appears, interestingly, to be a little bit higher in the females compared with the males, for reasons that's not really very well established. An incidence, as you might expect, is higher in the winter months, when often vitamin D status is lower and there are more viruses around. And of course, this illness often impairs athlete performance, so it's important that they can avoid it. It can actually prevent them from taking part in competition, or it can impair their competition performance and reduce their medal chances. Now, when it comes to probiotics, we've seen this slide from the previous speaker, or some variation of it, that probiotics can have potential beneficial effects on both the gut and the associated immune system. So for athletes, the benefits of probiotics could be the strengthening of the gut barrier function, or via modulation of immune cell functions. And we can see that this can occur in two different ways, through activation of existing immune cells and migration of those cells to other mucosal sites like the lungs. And this boosting of immunity, if you like, can improve our resistance to infection. On the other hand, we can also have anti-inflammatory effects induced by activation of T-regulatory cells secreting anti-inflammatory cytokines like IL-10, and that can actually improve immune tolerance of infection. And if we look at that actual paradigm, this immune resistance and immune tolerance in a paradigm of immune function, we can see that probiotics, which are the ones that are highlighted in blue font here, can have various effects that may improve resistance to infection. That would decrease infection incidence. In other words, the athlete gets fewer infections over a given period of time. However, some probiotics may also actually dampen immune responsiveness once a person does become infected through anti-inflammatory effects. And this can improve immune tolerance, the ability to dampen defense, but still control infection at a non-damaging level. And the consequence of improving immune tolerance is the infection burden is decreased, and we see reduced symptom severity and/or reduced illness episode duration. And as you can see, there's lots of other potential dietary components and supplements that can have these sorts of effects. Now, when it comes to actual evidence about whether probiotics work in terms of reducing the incidence or severity or duration of illness symptoms in infections, if we look at this recent meta-analysis, it's based on 23 trials. It's based on measurements made in children, adults, and the elderly, but actually excluded athletes. And most of these probiotic intervention studies were 3 months or more. And overall, the findings were that there was some benefit in using probiotics to prevent acute upper respiratory tract infections in this general population, and a likely benefit in reducing the duration of episodes. But as I said, this study didn't include any studies from athletes. They were purposely excluded because of the extra stresses that they endure as part of their athlete lifestyle. But there has been a fairly recent 2022 systematic review of 16 studies, all of which were on athletes, and examined the impact of probiotic supplementation on the risk and severity of infections, both respiratory infections and infections of the gastrointestinal tract. And if we just cut to the summary here, we see that the majority of studies reported beneficial effects of probiotics in reducing the risk of developing the examined infections or the severity of the related symptoms. So again, a suggestion that the probiotic is being beneficial in terms of both improving immune tolerance and immune resistance. And if we look at just some examples of the typical sorts of studies, they're generally small scale. They last anything from about 4 weeks up to several months. They tend to use this sort of dosage of a probiotic. Most of the studies have used lactobacillus probiotics, some on bifidobacterium as well. They often take place in the winter when, again, the infection incidence is the highest, and you're most likely to see a beneficial effect, if there is one, of a probiotics. And they're generally small scale. In this one, only 20 athletes completed the study. But you see, as you'll see, the results are fairly consistent. In this one, we see fewer days of illness symptoms and slightly, almost significantly, reduced severity of illness symptoms. And that was on runners. And this was one we did at Loughborough, where it was a mixed group of endurance athletes and games players, again using a lactobacillus probiotic, this time the Yakult probiotic, during 16 weeks or 4 months of winter training in the UK. And here we saw, again, a reduction in URTI symptoms, the number of weeks, the duration, if you like, of the episodes, and a reduction in the overall number of URT episodes in those in the probiotic group compared with the placebo group. Another quick example, one in male cyclists during, again, 11 weeks of winter training in this case. And again, a reduced number of URTI episodes in the probiotic group compared with the placebo group, and a reduction in the actual duration of URTI symptoms for each episode. So you get over the colds quicker on the probiotic treatment than you did on the placebo treatment. And then we have this International Society of Sport Nutrition position stand, which was published just a few years ago, that I was a co-author of. This sort of nicely summarizes the two main points I wanted to make during this talk. One is that athletes have varying gut microbiota compositions that appear to reflect the actual activity of the individual in comparison to sedentary people, in other words, their physical activity level. And also that's linked, of course, to the amount of exercise they do. And also it seems to be associated quite strongly with the amount of protein that they consume on a daily basis. And secondly, in an athletic population, specific probiotic strains can reduce the number of episodes and or the severity or duration of upper respiratory tract infections. And a third one here that we've not really mentioned, but related to that gut barrier improvement effect potentially of probiotics, is that intense prolonged exercise, especially when it's performed in hot environmental conditions, has been shown to increase gut permeability, which potentially can result in systemic toxemia. And specific probiotic strains could potentially improve the integrity of the gut barrier function in athletes. So what we see nowadays when we're making recommendations as to how athletes can help themselves to reduce their risk of infections, you commonly see the inclusion of a recommendation to take a daily probiotic, and perhaps also fermented foods as well, as part of their normal diet. And there are other sort of nutritional recommendations that we can include here. And then the rest of it is to do with sort of limiting or maintain or the training load, getting good quality sleep and that sort of thing. And of course, that all helps to maintain robust immune function, improving immune resistance and tolerance, which will help to reduce our risk of infection. And the other side of the coin, again, is preventing transmission or limiting transmission of pathogens in the environment into the body. And as we've all gone through the horrors of COVID over the last 2 or 3 years, we'll all be familiar with these sorts of general recommendations about how you cut down or limit the transmission of harmful microorganisms into the body. And finally, I'll finish on this slide, and this is something I've not touched on so far, but I know some of the other speakers are going to sort of delve into the more greater detail of this. But basically, it's about this interaction between the brain and the gut-brain axis. And we know that if you get physiological or physical stress, it can disrupt the colonic epithelium mucosal integrity in the gut. That could help to be protected against by the actions of probiotics in modifying the gut microbiota. And this in turn, taking probiotics, not only may affect immune function, but it may affect functions associated with the brain. So there are some studies, emerging studies, whether they're in animals or in humans, that are coming out that are suggesting taking probiotics may actually improve mood, it may reduce stress reactivity, and may also improve sleep quality. And if you think about it, all those three things, stress, sensations, the mood of the individual, and the quality of sleep they get are also things that would be very much of benefit to the athletes. But I'll leave it for others to talk about the details and the mechanisms that are involved in that. And I'll finish there, and thank you very much for your attention.
Including the meta-analysis, which is showing the effect of probiotic in a positive way in athletes. I would like
Yeah. Any questions from the audience side? Yes, please. Yeah. Take the mic.
there, like on the. I'm not sure whether he's online still.
Yes, thank you. Am I audible? You're
Looks
audible
like
here,
he's
but
not
what about
there.
Professor Gleason?
Yes,
No.
I can hear you now.
You are there.
you.
Yes, okay.
Thank
Yeah.
you.
We just we will take some question. Yes.
Since you mentioned lowering of the severity of illness, I just want to know if there is any idea about what IL-6 has to do in this, given the background of COVID, what IL-6 might be doing in the case of probiotic administration. Thanks.
I don't know the answer to that particular question, but I know that in response to exercise, IL-6 is actually increased during exercise, and it's actually being secreted not from immune cells, but from the contracting muscle fibers. And it does so in response to lowering of muscle glycogen levels. And this increase in IL-6, which is, of course, an inflammation responsive cytokine, which then induces a cascade of anti-inflammatory effects. It induces IL-10 release from your T regulatory cells. It induces more cortisol release from the adrenal glands. And these have a dampening effect. So this could be a mechanism through which doing regular exercise not only has many other health benefits, but in particular influences immune tolerance. And we've seen in the COVID pandemic, one of them, there's nothing you could actually eat during COVID that could actually prevent you from getting infected with COVID if you came into contact with an infected individual for long enough. But what we did see was that people who are fit and have a healthy diet and are not overweight have far less risk of having severe and life-threatening symptoms and inflammation of the lungs associated with the more severe outcomes of COVID. So there is a protective effect there that probably does involve IL-6 from a mechanistic point of view.
Can I say? So in fact, IL-6 helps in the immune cells differentiation, especially in the T and the B cells differentiation. So in a way that probiotic works, increases the IL-6 and it increases the T cells and B cells differentiation. So and boosts the immunity.
Okay. Thank you for the comment. So we will go to the next speaker now. Thank you, Dr. Gleason, for joining us and for the wonderful talk. Thank you so much.
Thank you very much. Thank you.
Thank you, Dr. Rama Chaudhary. Now I invite our next speaker, Dr. Namita Jaggi, to speak on the gut microbiome, antimicrobial resistance, and probiotics. Dr. Namita Jaggi is currently the chairperson Lab Services and Infection Control, and Chief of the Education Research at the Artemis Hospital, Gurgaon, India. She is the alumnus of the Maulana Azad Medical College and has more than 30 years of experience in microbiology and infection control. Her key research interests are microbiology, antimicrobial stewardship, and healthcare associated infections. Under Dr. Namita's leadership, Artemis Hospital was awarded the WHO Asia Pacific Hand Hygiene Excellence Award. She has also been the recipient of APIC Hero of Infection and Prevention. She is an executive member of this APSIC and Hospital Infection Society India and country secretary for INICC. Dr. Namita has numerous publications in peer-reviewed journals to her credit. She is also actively involved in translation research at Artemis by supervising several research projects and co-guiding DNB and PhD thesis. Now over to Dr. Namita.
Thank you, Professor Samuel Raj, for that kind introduction. Professor G.P. Talwar, Dr. N.K. Ganguly, seniors, juniors, students. It's a real pleasure to be here, and I thank Dr. Ganguly and Mirja for giving me this opportunity to be here today. So I think it's time to shift a little gears and go on to antimicrobial resistance. So I'd like to start with a little slide, and this is not moving. Can you help in this? Okay. So I want you to imagine a world without transplants, without cancer treatment, without joint replacement and intensive care. Seems a bit far-fetched in today's world. I want you to imagine a world without antibiotics, because that is the place where we are going. We are entering the post-antibiotic era. WHO predicts that in 2050, we will have more than 10 million deaths, which is equivalent to the cancer deaths, or perhaps more than the cancer deaths annually. And that's a very, very lingering, silent pandemic, just like the COVID, but it's a continuous pandemic. So here, I want to just pause on the why of it. And as you all know, it is because of the inappropriate use of the antimicrobial drugs in a One Health world, where we talk about the patient, the human, the hospitals, as well as the communities, as well as the environment, the animals, the veterinary, the agriculture, the fisheries, everything together. At a population level, it causes the antimicrobial resistance. And at an individual level, it causes not only side effects to that person, but also the ARG, the antibiotic resistance genes, they accumulate as a reservoir in the gut. And gut is known to be the epicenter for antimicrobial resistance. Now, if we were to find out a solution, because the problem is multifactorial, the solution is also multifaceted. And to prevent the emergence of resistance, we use judiciously the antibiotics in all sectors. And since I am an infection controller and a microbiologist, and we lead the antimicrobial stewardship in the hospital, we have about 16, 17 years of data of antimicrobial use, of antimicrobial resistance. We track the rates and trends of all the gram-negative and gram-positive bacteria. We have an antibiotic policy, and we have antimicrobial stewardship. But when you prevent the spread, we have to follow the policies of infection control. And part of fighting this spread of AMR is identifying reservoirs of resistant organisms in the environment, in the animal and human gut, which is talking about microbiome, as also the new drug discovery, which is really not under the purview of today's presentation. So we'll focus on the microbiome today and on alternatives to antibiotics in terms of pre and pro antibiotics. So this is a very, very simplistic view, especially to the gathering sitting here, but I just wanted to include it so as to just get a perspective. So we have antibiotics, which is opposing life, and we have probiotics, which is for life. And as far as 2000 years back, Hippocrates also said that it starts with the gut. So the gut is really, really important. The actual scenario is not as simplistic as this, and we have the host to contend with. So there are a lot of host factors. So today I'll be just taking a few introductory slides on microbiome, the effect of antibiotics on microbiome, that is eubiosis, dysbiosis, and rhizostome, which is the cornerstone of today's presentation, the mitigation strategies in terms of role of probiotics and prebiotics, and the future directions. So as we all know, the microbiome, which was coined by Joshua Lederberg in 2001, and since has had many changes of definition. And the beautiful definition is the microbiota plus the theater of their activity. I really love this because it includes everything, which is inclusive, all the metabolites, the peptides, the lipids, the polysaccharides, all the signaling molecules, toxins, etc. And since the Human Microbiome Project in 2007, this has gained popularity. So the research, and I was talking to Dr. Ganguly in the morning today, the research has been going on for many decades. And I was telling sir that probably 3 decades, and he was telling me, no, it is further beyond that. But since it picked up steam since 2007, this is just to show the distribution of the gut microflora, which is concentrated in the colon. The gut microbiota in adult humans is dependent, as we all know, on the diet, lifestyle, geographic location, the genetics which you have inherited, your environment, and antibiotic intake. And eubiosis, as we know, is a balanced and symbiotic state where we have the beneficial phyla, 90% of Firmicutes and Bacteroides, and low abundance of potentially pathogenic species like Proteobacteria, Enterobacteriaceae. This is the gut microbiota composition. As we know, the 2 phyla, Firmicutes and Bacteroides, include the 90%. And among them, we have the 95% being presented in Firmicutes by Clostridia. And we have less of Actinobacteria, Proteobacteria, Fusobacteria, and Vermicomicrobia. The Firmicutes Bacteroides ratio is talked about a lot. And I was speaking to Dr. Prajapati also, and he was giving me some Indian data, which is really very valuable. But in very, very simplistic terms, if the Firmicutes increase, it leads to obesity. And if it decreases and the Bacteroides increase too much, it leads to inflammatory bowel disease. Of course, the ratio evolves during the different life stages in the infants and the elderly. Also, the gut microbiota evolves in humans. So depending on whether a baby has had a cesarean delivery, where they usually inherit the skin bacteria, to a normal vaginal delivery, where they inherit the vaginal flora, rich vaginal flora of the mom. It also depends on whether the baby was born preterm or full term. And also the placental microbiota is also a cornerstone. And really, when I was going through literature preparing for this presentation, I realized that this is just the tip of the iceberg that we are really dealing with. And this science is really, really evolving. In the infant also, depending on the feeding mode, maternal diet, host genetics, family lifestyle, and geographic location, the gut diversity changes. And the first 2 days of life, we have more of Clostridia, Enterobacter, and Streptococci. And from the 4th to 7th day, the bifidobacterium starts predominating. And after the solid food introduction, the infant gut microbiota resembles that of the adult. Disturbances in the gut microbiome. Now, when the gut microbiome shows a resistance, the composition stays much the same. When it returns to its original composition, it is resilient. When there is a same function, it retains the functional redundancy. But dysbiosis is characterized by a state of irreversible change in intestinal microbiota with damage at the level of genes and proteins. And coming to antibiotics, which disrupt the gut microbiota homeostasis and causes dysbiosis. And dysbiosis is an alteration in both the composition and the function of microbiota caused by host-related and environmental factors that overwhelms the resistance and the resilience capacity of the microbial ecosystem with the accompanying symptoms of dysbiosis. So these are the effects of antibiotics on gut microbiota with reduced diversity, and it takes a very long time for it to come back. In children, it comes back faster in about a month. In adults, it can take 1.5 months to 6 months to years. And there is also overgrowth of pathobionts like toxigenic Clostridium difficile. There is altered metabolome. There are breaches in the intestinal barrier, decreased mucus and increase in inflammatory cells. And there is increase in the antibiotic gene burden, which increases the spread of antibiotic resistance and is known as resistome. This is just a diagrammatic representation which shows that the blues and the greens, which are the good bacteria, when they are exposed to antibiotics, it leads to a replacement by the pathogens and it causes infections, inflammations, and exacerbations. And ultimately, we have a gut full of these browns and these pathobionts. The effect of antibiotics is not the same for everybody. It depends on the duration, the doses, the antibiotic classes, the host age, the genetic susceptibility, lifestyle, and the individual's native microbiota. So it's not really the external, and the host is really a very important parameter in this. And this is showing how the antibiotics are modulating the gut microbiota. And in cases like fluoroquinolones, ciprofloxacin, and levofloxacin, restoration takes up to 1 year, whereas with lincosamides and clindamycin, the restoration takes up to 2 years. Beta-lactams, it takes about 2 months, and glycopeptides like vancomycin, it takes about 5 months. So it takes a long time for it to restore, but each class of antibiotic has a different effect. So resistome is a collection of the antibiotic resistance genes harbored by the microbiota. It can be intrinsic, it can be mobile, which is transferred by the mobile genetic elements, which we will be shortly talking about. And it can also be the cryptic or the proto-resistance genes, which are expressed only during horizontal gene transfer or some switch in the mechanism. The mobile resistome is harbored most abundantly by the Proteobacteria, Firmicutes, Actinobacteria, and Bacteroidetes. And the human gut environment is a very ideal environment for the horizontal gene transfer, where there is a relatively high selective pressure of the prophylactic or the therapeutic antibiotics, and this contributes to the global spread of these antibiotic resistance genes. We all know about the conjugation, transduction, and transformation, the methods, but we must realize that any damage to the intestinal epithelial cells by means of antibiotics, inflammation, reactive oxygen species, antibiotic exposure, and also biofilms hastens this process of acquisition of these antibiotic resistance genes. This diagram also just shows that the susceptible strains are wiped out by the antibiotic, and the non-susceptible strains are growing with promotion of the horizontal gene transfer. The factors affecting the gut resistome is the diet. And once we understand all these factors, we will be better able to fight against AMR. So the high fiber diet, that is the more carbohydrate diet, the high fiber diet, that promotes the growth of the better bacteria. Use of antibiotics promotes the resistance transfer. Eating meat from animals harboring the ARG continues to put the ARG into the body, and also consuming produce contaminated by manure containing the antibiotic resistance genes. Animals to human transfer has also been seen very frequently. The environment, the hospital environment, the antibiotic factories environment, and the animal feces, all this leads to a lot of gene transfer by the horizontal route. Geography and tourism is very, very important, and every geography really has a signature microbiome. And we are the most mobile genetic elements. We are moving from one place to another. We are traveling, and that is leading to a lot of spreads. And of course, the antibiotic as a pressure. The molecular diagnostics, since the molecular diagnostics have come up in terms of metagenomic sequencing, in terms of functional metagenomics, in terms of genome-resolved metagenomics, and in the metagenomic chromosomal confirmation capture method and the long-read sequence technologies, all these have really helped us in our fight against the AMR in the AMR context in order to read the microbiome genes. We have these ARG databases, and now earlier we used to have the antibiotic resistance gene database as ARDB, but now we have the comprehensive antibiotic resistance database, which is essentially a bioinformatics tool and consolidates the genomic data. We also have antibiotic resistance gene annotation softwares, and all this has really also helped us to study the antibiotic resistance genes better. And now Resistome Map is an interactive display set up for the complete visualization of the gut resistome. The next part of my presentation is on possible antibiotic alternatives. Now, we are talking about phage therapy. We are talking about bacteriocins because the antibiotic pipeline is basically dry. We are not getting any new antibiotics very soon. So we have to look at alternatives, and the bacteriocins, probiotics, fecal microbiota transplant, which Dr. Sareen was talking about in the morning, and the monoclonal antibodies are coming in a big way. So since we are talking about probiotics, which are live bacteria, live microorganisms, the prebiotics and the symbiotics, which are a combination, is under focus. And basically the strains which are used are lactobacillus and bifidobacterium primarily. Others like Saccharomyces, Streptococcus, Enterococcus, and E. coli bacillus. And they're usually about 1 to 10 billion CFU per dose, but some can go up to even 50 million. Now, probiotics, as we all know, they need to be alive. They have to be less pathogenic or lack of pathogenicity. They have to tolerate. They have a capacity for adherence, and they need to have a health benefit and immunomodulatory capacity. And this diagram is a big favorite because I think all the speakers have shown this. But in terms of antimicrobial action, the first one is the enhanced barrier function because they enhance the barrier function. They secrete antimicrobial compounds themselves. They modulate the immune function, they competitively exclude the pathogens, and they block the adhesion of the pathobionts to the target cells. The role of probiotics in human health as a dietary supplement is explored and accepted by the FDA as an alternative to antibiotics. There have been large meta-analysis and a lot of studies, but it is not yet accepted by the FDA. Probiotic alone and a synergism, that is a combination with the antibiotic. These are some of the probiotic microorganisms which have been studied in various studies. There are some of these studies which I found, which is where probiotic instead of an antibiotic has been successful. And many of these studies are in periodontal biofilms, in the periodontal health, in periodontitis, and in Candida albicans infections. Currently, there is no standardization for the clinical use of probiotics as an antimicrobial therapy because the definition of dosage, mechanism of action, and clinical efficacy remains to be determined. However, the probiotic use in livestock agriculture of chickens and turkeys has helped replace antibiotics in preventing disease like salmonella, decrease incidence of diarrhea, and promoting growth on livestock animals. This is a very interesting study about the role of probiotics as quorum quenchers, which must be explored, where the pathogen elimination by the probiotic bacillus via signaling interference, it's a very interesting, where Staph aureus colonization in the gut and nasal was not detected when the intestinal bacillus was present. Another study, probiotic disruption of quorum sensing, reduces the virulence and increases cefoxitin sensitivity in methicillin-resistant Staph aureus. The safety of probiotic. I know one of the speakers in the morning also talked about the safety and the antibiotic resistance genes which are present, can be present on the probiotics also. So we have to be very careful. No unified World Health Authority has taken full responsibility. We have the European Food and Safety Association, which gives the QPS and the generally regarded as safe grasp by the US FDA. And QPS is the one which is more acceptable. So I think a lot of countries which don't have these regulations must include them. The lactic acid bacteria, Lactobacilli, Enterococcus, and Bifidobacterium, I'll just take a word about them. So lactobacilli have been extensively studied. They inhibit Helicobacter. They do not produce any disease in humans, and they have intrinsic resistance to some of the antibiotics like bacitracin, kanamycin, teicoplanin, vancomycin, and beta-lactams. And this may be an advantage if we are using an antibiotic-probiotic combination in the future. Enterococcus can be opportunistic, and so we have to be careful with it. And the Chang et al. In 2007 demonstrated that some commercially available food and drugs with probiotics contained lactic acid bacteria positive for antibiotic resistance genes themselves, located in mobile genetic elements, and they had the GRAS status. So we have to be extremely careful. Bifidobacterium inhibits the adherence of enterotoxigenic E. coli, enteropathogenic E. coli, and Clostridium difficile. And again, they have an intrinsic resistance to many of the antibiotics, which may be an advantage if an antibiotic-probiotic combination is used. This is the probiotic-antibiotic synergism, and there are many studies to show where they have been added with antibiotics to give a synergistic effect. The effect of probiotics on resistome needs further studies because one is that they can reduce the number of the antibiotic resistant gene in colonization permissive antibiotic naive individuals. But sometimes they exacerbated the antibiotic mediated resistome expansion after the antibiotic was given. And also that the stool sample does not completely correlate with the status of the resistome, and there is need to use the endoscopic samples. More studies are required on person-specific and antibiotic-dependent effect of probiotics on resistome because the host may exert an influence as also the antibiotic. We must also consider the ecological context in which probiotics are supplemented when the colonization is resisted by the microbiome of the host itself. Then the probiotics do not elicit an effect on the microbiome. Probiotic use is, however, clinically proven to modulate infant gut microflora disturbance after antibiotic use. This is a very interesting study in Nature. It's a very recent study, 21, where probiotics impact the antibiotic resistance gene reservoir along the human GI tract in a person-specific and antibiotic-dependent manner. These are certain bioengineered probiotic or pharmacobiotics, which is a very interesting concept, where we orally administer these pharmacobiotics and they go inside the gut and they have antipeptide activities. They're genetically engineered. And this approach can hold great potential in the coming years. And these are some of the engineered probiotics studied for diagnosis and therapy. The way forward, so this is my last slide. In diagnostics, the way forward is to develop microbiome-directed diagnostics to characterize and quantify the role of gut as a reservoir for ARG carriage, emerging antibiotic resistance and exchange, and aid in the development of therapeutic and preventive strategies to combat AMR. It can also be a very powerful tool for outbreak tracing. Regulatory guidelines, safety guidelines need to be established in all the countries. And in a clinical perspective, the actual role of probiotics in a person and antibiotic dependent way should be considered. Genetically engineered probiotics are going to be coming in a couple of years, and the increased role of microbiome in precision medicine. So with that, I end my talk. Thank you.
Thank you, Dr. Namita, for your excellent presentation on the gut microbiome, antimicrobial resistance, and probiotics. And you started with an excellent way of saying that when it's an alarming situation where we are going towards. We cannot imagine that the world without the antibiotics. And as you mentioned, it's very clear that more than 10 million deaths only due to this antibiotic resistance. And when we are talking about only about the AMR, so what are the possible ways to tackle it, or how we can mitigate that one in our CS, given a very clear indication that, what is a possible alternatives to the antibiotics? And one of the alternate she was talking about is the probiotics with various strains that, she has given an overview. Considering the time, can we take one or two questions from the audience? Quick, one or two questions. Yes, please. Yes, yeah, please.
I have a very simple, practical question for you. Recently, we had this H3N2 outbreak in Mumbai and many other states, I think in India, and it has been really bad with a lot of bronchospasm, fever, cough, like you get up breathless and they're being hospitalized, especially children and 50 plus adults. Now, you have mentioned, however, whatever we may say, the real life experience is if your near and dear one is not well, you start an antibiotic because you're scared. You know you're a doctor. It has happened in doctors' houses, but it doesn't happen that way because you try to give an antibiotic irrespective. Now, the point is that I liked your probiotic antibiotic combination of the pharmabiotics, and we do not know which antibiotic, which probiotic, what is happening. We exactly are nowhere actually at the current moment because research is far from happening in a very crucial way. Now, what do you suggest? What happens? We need to give antibiotics as well. Do you think it has been diminishing the resistance from before? We should talk a lot about antibiotic resistance before. Is it coming down? Or are we right enough adding a probiotic, any probiotic, be it a lactobacillus, a spore, or a fungi? So I would like your comments on that, being a microbiologist. Thank you.
Thank you, ma'am. It's a very, very interesting question as well as very complex. It's actually it's $1 million question. If somebody were to answer it today, I think they would be, really the king and queen. Okay, so to answer your question in two parts. The first part which you asked, is antibiotic resistance decreasing? No, it is not decreasing. Unfortunately, it is not decreasing because of the misuse of the antibiotics in various sectors. What has happened with the introduction of the Global Action Plan and the National Action Plan across in India as well, the awareness about antibiotics is definitely increasing. But that doesn't automatically translate into judicious use. But yes, one thing is happening. Most of the hospitals have an antimicrobial stewardship committee in which they are trying to reduce the non-judicious use of antibiotics. It is happening more in the ICUs and in the hospitals. So there is a controlled antibiotic, giving the antibiotics to the person. But in the OPDs, it is not that okay. Second thing is, in the metropolitan cities, it is still controlled. But as soon as you go outside of the metropolitan cities, especially in the smaller cities of India, it's the pharmacies which are prescribing the medicines. So there is less of in the community. So these are the untapped areas really where one really has to do research. In the communities, the people themselves are taking leftover antibiotics. So the problem is, I would say very, very alarming at this stage. So on one hand, we have to stop the emergence, like I said, by promoting the judicious use, and we have to do everything possible for that. That means in the hospitals, we have to take steps. In the communities, we have to take steps. And we have to even include the patients who are the ultimate users as groups. And on the other hand, we have to have the alternatives of antibiotics and research into that. Which brings me to the second part of your question, is talking about whether we should give probiotics. Now, yes, probiotics should be given. These days, the pediatricians and the intensive care people and the medicine are all prescribing probiotics after a course of antibiotics, that is after the course. And now we have research into the synergistic. So that is also getting built up, and there are numerous studies. But you're right, absolutely, that we don't exactly know what is fitting. But yes, we are taking a shot in the dark. For example, we know that lactobacillus, bifidobacterium, these are the useful probiotics. But again, here, the host factor, it may work very well in you and it may not work in me. It may work very well maybe for a person in the US, those particular strains, and may not work in India. So the geographical location, your genetics, your particular comorbidities in your body, your particular genetic signature, everything is going to affect it. But that doesn't mean that we should not try. So very simply put, yes, with antibiotic use, first of all, do not give when not required. If you have to give, yes, follow it up with a probiotic. I hope that answers your question.
So,
Thank you.
Just add a comment to it. Very well answered, Dr. Jaggi. So what Dr. Lahiri asked was the combination. I think it's another aspect to that is that many of the broad spectrum antibiotics when given leads to antibiotic associated diarrhea. So somewhere the combination with lactobacillus prevents that.
Yes.
And if you develop C. difficile infection, you are going to give vanco and then emergence of vanco-resistant enterococcus. So you are in that vicious cycle. So therefore, adding probiotic or lactobacillus to these broad spectrum antibiotic, especially amoxicillin and others, definitely is a good step forward. And yes, the strains and ability that one needs to study further.
Thank you.
There is a question from
Thank you. Dr. Narendra Mehra, sir.
Thank you very much for this wonderful talk. I really enjoyed it. Wanted to know that you use the word One Health and the horizontal gene transfer. Very important. I wanted to know the factors that influence the horizontal gene transfer. Is it genetic susceptibility, AMR, host immunity, all of the above, or is it worked out?
So from what I know, and I think this field is just starting to get evolved, but from what I understand, horizontal gene transfer takes place in reservoirs. And the reservoir for us is the environment. It's the gut of the animals, because we are talking about One Health, and also our gut. So the horizontal gene transfer is actually influenced, what you said, the influencers, it is external and the internal. So it's the host as well as the external stimuli. In the external stimuli, the antibiotics is the topmost thing which is going to stimulate the horizontal gene transfer. Also, where a lot of bacteria are present together, it is going to happen. And the internal, again, it is the host. So the host factors are relatively yet again understudied at present. Sometimes people may have a lot of horizontal gene transfer and others may not have. I think Dr. Ganguly sir wants to make a comment on this.
Sir. Really, this happened both the transverse and horizontal transfer because the grazing area for animals and the space between animals and humans, whatsoever it is, whether it is rhesus monkeys or cattle or anything, is shrinking. And a lot of antibiotic use in the food and other stuff, they create resistomes within the animal. And they then both, they have the same flora sometimes populates the human, and sometimes within the human and the animal guts, the strains, the gene transfer occurs, and then the humans start carrying that. Absolutely. And few of the experiments which have been done in One Health, one of the best is the Mycobacterium bovis and Mycobacterium avis complex, which is rampant in cattle and is transferred to human. And also recent studies have shown that some of the drug resistance, which are genes which are transferred from these bacteria to human strains is happening. This has been done with the Penn University in India, and one of the suggestions they have given is that they're given BCG vaccination. This study is happening in both Africa and India. So both things are possible. And genetic susceptibility is this, that if it is happening in children, but not all the strains are similarly, even if they're carrying the resistance gene signature, they are not similarly causing septicemia. Yes, there are some additional factors in them which people are studying. There's a very good study by Sulagna where she has shown that in the neonatal sepsis, the strains which are in the which carry rhizostomes which are in the skin of the child has not caused the septicemia. That is another strain.
Another strain. There was a study like what you were saying, overcrowding, like what sir was saying, overcrowding. There was a study in the veterinary students. One study I was reading about where they did the microbiome analysis and they found them to carry antibiotic resistance genes, and they had never been exposed to antibiotics. So therefore, it is the vicinity, it is going into the soil and then going into the feces and then handling. So it's a very complex cycle, and that is why I think it is One Health. So we cannot only tackle the human part of it and feel that we can tackle antimicrobial resistance.
Yeah, thank you very much.
Thank you.
Dr. Namita, it's really... Dr. Nirja has a question. Yeah, please.
Post-antibiotic intervention. You were saying the post-antibiotic intervention, it's quite possible that you lose certain species of bacteria. And there are some species, I was reading a paper, where there are certain species which are lost forever and don't even come back after. So do you think an intervention like a probiotic or whatever, symbiotics, you think they could be useful in getting back those lost species?
So in the probiotics that we are dealing with, we have about 7 genera that we are dealing with, and most of them are beneficial, like the Lactobacillus, Bifidobacterium. So I don't think they will get back the lost ones. But once you remove the selective pressure on any particular, antibiotic, and you have selective pressure for the bacteria and you have the better bacteria over there, then sometimes it has been shown that the antibiotics which we were using, maybe 10 years back, sometimes, they are going to be, like, for example, the cotrimoxazole. cotrimoxazole, Septran. Septran is an antibiotic which we used to use a couple of years back, 10, 15 years back. Now, when people stopped taking that antibiotic and the selective pressure went out on that, sometimes they come back and they become more effective. But in your probiotic, whatever 7 genera are there, they are going to inhabit. And of course, they are going to cause the beneficial effects. And they are going to get the good population of bacteria back. And in children, it has been really shown to be really helpful. And I think in humans also, in certain, like ma'am was saying, in Clostridium-associated diarrhea and many other conditions, I'm sure in the coming years, we will have the probiotics being used instead of antibiotics and with antibiotics also. So they're going to be having good effect.
I know that many hands are raising from the backside, but considering the time, we will not be able to take any more questions. Thank you so much, Dr. Namita, for your excellent presentation. And it's true that we need to go for this One Health approach. Thank you so much.
That was quite a remarkable session indeed. Can we have a round of applause for Dr. Namita Jaggi here for giving us her insights on such an interesting topic indeed. I would now be requesting our chairperson, Professor V. Samuel Raj, to kindly come forward along with Professor Rama Chaudhary as well, to kindly felicitate our dignitary here. Ma'am, please accept this token of appreciation, this token of respect from our end. Everybody, can we please have a round of applause? Thank you, ma'am, once again. Taking the lead once again by our chairperson here to call upon our next speaker in the session herewith.
Thank you very much. Our next speaker is Professor Lynn Barker. She will be giving the presentation on the gut-brain axis and the microbiome. Time to look beyond the brain in neurological conditions. Dr. Barker is an associate professor of cognitive neuroscience at Sheffield University, Sheffield, UK. She is an expert in the effects of the brain injury, stroke, and dementia on cognitive function and behavior. Her work has identified novel biomarkers of repeated conclusive events and developed new digital tools for cognitive assessment in neurological groups. She has received and been nominated for several prizes and awards. She is presently conducting several studies investigating the gut microbiome and potential therapeutic effects of probiotics on symptoms in Parkinson's disease, multiple sclerosis, amyotrophic lateral sclerosis, and long COVID. Now over to Dr. Barker.
Hello, India. Can you see me? I'm going to start to share screen, so hopefully this is going to work. Okay. Can we see the presentation, everyone? Yeah? Okay, I'll get started. Hello, everybody. Welcome to my talk. Thank you for inviting me to present some of our current and ongoing work. It's a real privilege to be here, and I hope to be visiting India next year, so to build partnerships and share knowledge. So do please keep in touch. I am, thank you for the nice introduction, an associate professor in cognitive neuroscience. I'm not a gastroenterologist and I am not a biomolecular scientist. So my interest in the gut microbiome is from its potential role in neurological conditions, and we're learning a lot more about that at the moment. I've spent the past 3 years learning as much as I can about the gut microbiome and the gut-brain axis around all my other work and research activities. And we've drawn as much as we can from our own data and existing studies about the gut microbiome and the gut-brain axis. We are partnered with our regional hospitals and 3 research centres, one of which is a centre of excellence for motor disorders. Our external partner, as you can see on this initial slide, is Symposium, who are a probiotic manufacturer. So I'm just going to give you a little bit of background to the gut-brain axis, but I'm sure you may know some of this already, but just to introduce the topic. The gut-brain axis was first discovered in the 1960s when it came to light that brain neurotransmitters were synthesized in the gut. It is a bidirectional signaling system. So what this means is that obviously our gut is in constant communication with our brain and vice versa. Not just about appetite and satiety, but also in terms of the endocrine, immune, autonomic signaling as well. Importantly, we now know that gut microbes also exploit these signaling systems. In addition to these biochemical pathways, there is also a physical route that connects the brain to the gut, and this is via the vagal nerve. So you can see on this illustration on the slide. The vagal nerve, as its name describes, the vagabond nerve which meanders through the body, innervates viscera and other internal bodily organs. One early theory that was proposed by Braak and Braak is that metabolites of gut microbiome, of the gut microbiota and other products, including misfolded proteins, can traffic along the vagus nerve and terminate at the dorsal vagal nucleus within the brainstem. From there, these products are able to proliferate across the brain without any impediment because they don't need to cross the blood-brain barrier. So they have unrestricted access to the brain. And this mechanism is now considered to be one of the ways in which misfolded proteins might accumulate in the brains of those with neurological conditions. So the enteric system, as I'm sure you all know, there's been lots of really interesting and informative talks, is a complex blend of motor and sensory neurons. And there is a general assumption that there are as many neurons in the gut as there are in the actual brain. So here I've just pinched an infographic slide from the APC unit at Cork in Ireland because I like it because it gives a nice little summary of information about the microbiome. And what this tells us is that there is now an assumption that 90% of diseases states can be linked back to the gut and the gut microbiome in some way. We have more microbes on our body, not just in our gut, than there are stars in the Milky Way, which means that we can be classified essentially as superorganisms. We each have a unique fingerprint of gut microbes, but more recently we are recognizing that there are what we call specific signatures. And a signature is a pattern of species, and it's usually species level that we're interested in when we talk about signatures, that is associated with certain conditions and also in health. The gut is filled with microbes, viruses, fungi, and archaea. We don't know really what the archaea do. They are evolutionary ancient bacteria that have been around for millennia, as long as we have. So just to give you a little bit of background there. In this slide, I just wanted to illustrate the phylogenetic tree when we're considering microbes. I'm sure most of you are probably familiar with this. So most analyses often focus on the phylum level, which is at the very top. So where you're talking about the general grouping of the microbes and the clusters that they form. But actually the detail is down at the bottom level in terms of the species. Now, a lot of studies don't focus on species level data, primarily because it's so complicated, it's so time consuming. Depends on the type of sequencing you use, but we could say maybe we can track up to 300 different species, maybe more, depending whether you're using metagenomic shotgun sequencing or 16S, and that is per person. So to chart those data, it takes a lot of time, but this is what we have been focused on in our research, looking down at the very detailed level of species level data. Here I've used a little slide that's taken from Wang's lab at Stanford, and what this shows is the microbe burst of our gut microbiota, and it's stained with an immunofluorescent dye, and it just gives you a perspective. I think that's only a 1-inch slice from the gut lumen of the amount of the population and the community inside of our guts. So one question is, why does it matter? Who cares? Yeah, we've got microbes in our stomach. Why is that important? Well, we know that gut microbes at the very basic level, if we want to talk about their importance, is that they metabolize short-chain fatty acids. We are unable to metabolize short-chain fatty acids ourselves. We need our microbes to do that for us. And that's really vital because the short-chain fatty acids are the ingredients that keep our gut intestine integrity safe. So it prevents leaky guts. Leaky gut is an issue because it can be a start point then for what turns into a dysbiotic gut. And as we know from all the previous presentations, that includes a range of different problems in terms of intestinal function. But the upshot of that is that those problems with intestinal function then affect other elements of the system, including individuals' neural function. So here's a little slide which just summarizes some of the conditions and problems associated with dysbiosis, and also the benefits of having a healthy gut, which is a eubiotic gut, which is on the other side of the slide. So we can see that in a dysbiotic gut, there are a potential whole range of diseases that might emanate from that original start point, and these are known to be diabetes, cancer-related, obesity-related, the general discomfort and problems associated with ongoing diarrhea and constipation, because the knock-on of that is that you're not going to be absorbing your nutrients properly, either from your food or your medication if you've got a neurological condition or another condition, and cardiovascular disease. Indeed, the list is endless. This is actually quite a truncated list. But what you might be asking, that's all well and interesting, but how does that relate to neurological conditions? Well, very early on in James Parkinson's first diagnosis of what he called the shaking palsy, he noted that many of his patients had gastrointestinal problems. Usually it takes the form of constipation, but I've spoken to a lot of our patients and actually that picture is more varied. They all complain of having some intestinal problems early on in the phase of the illness. Sometimes that can be up to a decade before the condition is diagnosed, sometimes longer than a decade. And so the theory is that the problems that then become the symptoms of Parkinson's disease have a potential start point within the gut. The theory is now that the microbiota becomes dysbiotic very early on. Now, what we don't know is exactly what's happening between the stage of the microbiota becoming dysbiotic and the symptoms of Parkinson's disease and other neurological conditions beginning to appear. But we do know that it seems to have something to do with the signaling pathways. Most of this is from animal data, some of it via the vagal nerve and some of it via the chemical and immune system signaling pathways. Of course, symptoms vary across individuals, but for one start point, if this can be improved in people with Parkinson's disease, not only do they have a better quality of life, because one of the things that they report is inability to enjoy meals with friends and socializing, and also problems with medication absorption. So we may be uncovering some of the mechanisms involved in the development of the condition, but we also may be contributing to improving the lives of those who suffer with these conditions. So if you remember my earlier point about a signature, there is some dispute about signatures and fingerprints, but I think as more data emerge, and definitely at species level, if species level data are carefully mapped, we do see signatures associated with specific conditions. And then around that signature will be the individual's unique fingerprint in terms of their microbiota constituent components. There is a study that's been conducted with over 9,000 people that's revealed that there is a distinct gut microbial signature that is associated with healthy aging and survival in the latest decades of life. And this is really informative because if we can get to know and understand what is a healthy microbiome, then we can also learn more about what's a dysbiotic microbiome. So what do we mean by a signature in relation to neurological conditions? And can we say that there is one associated with Parkinson's disease? So we conducted a small feasibility study with a small group of patients with Parkinson's disease. One group was a probiotic group and the other was the placebo group. We used a probiotic called Symprove, and we resourced all the probiotics available and chose that one because that one has the strongest empirical evidence that the microbes survive, make it to the gut alive, which is a problem for some of the probiotics claims, and then once in the gut can thrive and survive within that community. That's a very trepidatious journey for microbes, and many proposed probiotics, the microbes are already dead on the shelf. So what we did is we assigned our patients to either the probiotic or placebo group. We took a fecal sample at baseline and we took a fecal sample at 12 weeks, which is the duration of the course of the probiotic Symprove. Symprove manufacture their probiotic fresh on a daily basis, so you can't buy it in shops. It has to be delivered to your home, but that means that we know that we're getting a product that's got live bacteria in it. What we found, we dug down and analysed species data, and we found that there does indeed, in our group at least, seem to be a signature associated with Parkinson's disease. And this constitutes about 100 species. The groups were not different at baseline, which is important in terms of the profile of this signature. So the prevalence of these certain species were not different in the two groups at baseline, but we did see changes in prevalence and abundance between the two groups at 12 weeks. I'm going to show you some data now, which has not been published, but we are in the process of publishing that along with some other findings. So if you please not reproduce that, but I'm happy for this talk to be recorded. So we use these probiotics that are safe. They've got quite a lot of evidence, and we've conducted several studies with other partners as well at UCL in London, using ProDigest, which is a mechanistic approach using an artificial abdomen where you can apply the product straight onto the microbiome and you can assess the results within 24 to 48 hours. So we were very confident that we were using a safe product with our patients. And we were hoping that what we might be able to show is that we could ameliorate some of the problems that they have with ongoing intestinal issues. So this is a pretty complicated slide, so I'm just going to talk through it so you can understand what this is. You can see that this is the placebo data at time 1 and time 2 for the placebo group. These are the collection of species that we call the signature, in that they are present either 100% or down to 58% of our cohort, so above average. The light green is at baseline and the darker green is at 12 weeks. So what we did is we classified each of our species on whether they were considered beneficial, commensal, or whether they were considered to be pathobionts. This took a tremendous amount of work, but fortunately most species have at least one journal article dedicated to them. So we had to do that and cross-rate and make sure we're getting those categories right. But of course, there are contextual variants. The microbiome is a community, so sometimes whether a microbe is beneficial or commensal or a pathobiont will depend on the other microbes that are present in that community. So this is a start point for looking at data in this way, and we hope that other people start to do this, because I think it's really important for really being able to fully understand what the species are doing. So the red ones are the ones that we classified as pathobionts, i.e. you don't particularly want those flourishing. Bear in mind, this is only a small snapshot of the actual individual's microbiome. These are just what we call the signature data. So we can see in our placebo group that we've got a lot of red columns, which are the pathobionts in terms of the microbiome. And we can see that they either increase at the second time point, 12 weeks, some of them slightly decrease, but not many, and some of them just stay the same. What we thought this looked like is very much like almost the microbiome, the signature microbiome in our Parkinson's patients, was on a trajectory of decline, i.e. it was constituted by at least half not beneficial microbes, and that these microbes were kind of remaining stable or increasing in prevalence over a 12-week period. The brown category, by the way, are microbes that we couldn't really make a decision whether they were beneficial or pathobionts based on the literature around that species. The white ones are other, they are unclassified. But when we looked at our probiotic data at 12 weeks at species level, just for signature alone, we can see something quite different emerging. And this is only a relatively short time frame. So what we really want to do moving forward is look at longer time frames and see if we get more significant change, particularly focusing on the signature. What we found is that, as you can see with the greens, which are the commensal bacteria, they either remain at the same level. That's a good thing. So neither increased, but they didn't decrease either. In some cases, they did increase. And what happened with the pathobionts is we saw a general decrease in those over the 12-week period. So what this shows us is a complicated picture about what's happening in the microbial community. It's not simply a matter of dropping in some beneficial microbes and expecting those to proliferate and dominate. What's happening here is we're seeing subtle changes in a whole community of microbes in our Parkinson's disease patients, which seems to be a slowing of what is potentially a negative trajectory of decline that we see in the control group. So this is just a quick summary of some of the differences that we see. The control group had diminished prevalence of beneficial bacteria after 12 weeks. Bearing in mind the control group have not been interfered with in any way. They've basically taken a sugar supplement that's packaged the same as the probiotic, and they were blind to whether they were in the probiotic group or the placebo group. But what we are seeing in them over a 12-week period is as interesting as the findings that we had with our probiotic group, because we're seeing that there is this very negative change to what's happening in a 3-month period in the microbiome of people with Parkinson's disease. In terms of our patient group, they either increased in the prevalence of the beneficial bacteria within the signature or remained stable. We did get a few where there was a decline, and we would have expected that, because otherwise the data wouldn't have seemed authentic. We wouldn't have expected all of the beneficial microbes to have increased in prevalence. So to summarize, the gut-brain axis has a potential role in many conditions. As you already know, eubiosis and dysbiosis are important clinical markers, but we're only really just beginning to understand how important they are for neurological conditions. Evidence-based treatment should include prebiotics and probiotics, but also all the other things that we know that are really beneficial. For example, we know that being out in fresh air can have positive effects on the microbiome. Exercising has positive effects on the microbiome. Good sleep quality and relaxation also. Just to summarize some current studies that we've got underway that were part of the introduction, we're now looking at multiple sclerosis. This is a small feasibility study, and we're using a probiotic. We're using the same sort of study design, taking microbe samples at baseline and at 12 weeks follow-up. We've got another study looking at amyotrophic lateral sclerosis, and we've just started a study with long COVID patients. So my take-home message is that the microbiome is in direct and constant communication with the brain and vice versa through several pathways. We need more information and we need more research with human participants with neurological conditions to try and understand these mechanisms better, drawing from animal models and experimental data. Dysbiosis to the microbiome may actually have an effect on neurological conditions. It may be a causative factor. It may be a biomarker. That's yet to be determined. Probiotics can deliver healthy gut bacteria, but we need to consider how they do that within the community of the microbes, not just simply assuming that we can drop in several beneficial microbes and that we're going to see a proliferation of those microbes. What we're going to see instead is probably just a shift in the whole community. Our patients who were in the probiotic group but didn't know that they were in the probiotic group all reported feeling significantly better during their time taking the probiotic. And this included having improved bowel habits, being able to go out and socialize more and eat with family and friends, improved mood state and better quality of life. So research in human participants must continue in this area, but also we need to expand it to other conditions. And finally, there are just a few publications that we have at the moment, but we've got several in progress and under review. My book will be out this year also, so do please keep in touch. You can follow me on Instagram or you can drop me an email. Thank you.
Thank you, Dr. Barker, for nicely explaining the gut-brain axis and also explaining about gut talk, why does it matter? And also correlating this gut microbiome with the mental health and how gut microbiome is linked to the dementia, Parkinson's disease, and multiple sclerosis. If there is any question from the audience side, we can take a question. Yeah, Professor Ganguly. Where is the mic? Yep. Please give it.
Actually, a very nice study. I have a comment and I have a question. The comment is that in Luxembourg, there is an issue which only studies Parkinson's disease. The 400 scientists only try to find a solution to this. So they had they have also a very strong stool biobank in Luxembourg. So they were doing about 16 LC-MS machine where looking at the metabolome and other signatures. So with the probiotic intervention, some of these metabolic signatures improved in Parkinson's cases. But what I want to know from you that that there are studies with the green tea, the epidemiologic studies with the green tea, and if you give it to about 28 cups of green tea to the people, when this study was done in the Netherlands, there is a benefit in the cardiovascular outcomes. So could there be a study where we know that populations which take fermented food and others have lesser Parkinson's disease? Is there any epidemiological evidence to that? Or if I want to restore the flora in Parkinson's disease, should it be upper gut or the colon?
Right, thank you for those comments. I'm not sure how well I can answer them because I'm not a gastroenterologist. But my feeling is that we already know that positive changes to diet has a positive effect on the microbiome. I'm not familiar with the green tea project, And I don't know whether there are any epidemiological studies on Parkinson's disease. I know that for type 1 diabetes, for example, there's far less incidence of that in Asian countries, which seems to be associated somewhat with diet. But I don't know for Parkinson's disease. I think the prevalence remains fairly constant as far as I'm aware. But I need to look at epidemiological data to be able to answer that one.
Thank you. Thank you for your nice study. The two important factors that we know that affect Parkinson's is the genetics and environment. In your study, did you also take or recruit Parkinson's with the familial phase? I mean, patients who had a family history positive and the family history negative, and then use of probiotics?
No, we didn't, because it was a feasibility study, but we are going to hopefully conduct much larger studies. We've got several grants under review and under application to build upon this, but that's a really useful suggestion. No, we haven't done that. We haven't got a geneticist on the team, and that might be useful to consider.
And the other factor is the autoimmunity, because Parkinson's disease is also one of the autoimmunity bases. So if you would look for the antibodies, autoantibodies, and then see those who have higher or lower levels of autoantibodies.
Yeah.
The effect of probiotics on them.
Yeah, absolutely. And we would expect to see some effect, wouldn't we? I mean, we would hypothesize that there would be, particularly if the probiotics had known anti-inflammatory properties. So yeah, absolutely.
Yeah, thank you, Dr. Barker. And this is the last talk for this particular session. Thank you so much.
Thank you.
Thank you.
Bye-bye.
So we'll conclude the session now for today. Thank you so much.
That was quite an impressive session indeed. And we are really thankful to both our chairpersons here for diligently conducting the session forward. I would now be requesting the humble presence of Mr. Takeshi Yoshimoto, sir, to kindly join us here on the stage and felicitate our chairperson, starting with Professor Rama Chaudhary ji. And also in the meantime, let's welcome Mr. Takeshi Yoshimoto with a round of applause, please. Hands up in the air. Yes. Thank you so much, sir. Thank you, Professor Rama Chaudhary ji, for giving us your precious time here today. Please accept this token of appreciation from our end. Thank you, ma'am. And thank you, sir. I would also request the humble presence of Mr. Junji Fujimoto to kindly come forward here on the stage and present the token of appreciation to our chairperson, Professor V. Samuel Raj. Everybody, can we please have a round of applause? Thank you. Welcome, sir. So please accept this token of gratitude, this token of respect from our end. Thoroughly delighted to have you here with us. Thank you so much. We now move on to one of the most exciting parts of the symposium, which is the panel discussion. The theme of the panel discussion is probiotics as vaccine immunity boosters in immunization. We would now like to call Professor N.K. Ganguly to moderate the panel, and I request sir to kindly take the panel discussion forward. Can we welcome Professor N.K. Ganguly, sir, with a round of applause, please? Welcome, Mr. Ganguly.
Actually, this session is meant for our younger population who are sitting at the back to clarify some of their ideas about the probiotic and immune responses. So I will request the panelists, a very distinguished panelists with years of experience. They will have two questions each for, and we'll have 4 or 5 minutes to explain. But since they're very experienced, their skill will enable them to do that. But you could later on ask more clarification from them. But we are changing the format a little bit, that I had about 5 minutes, which I will forego, so they can create a question for them by themselves and say it. So they will have 3 questions actually. And then there will be enough time, half an hour, to ask them about the clarification. So this is not for the learned group. This is for those who are young and who want to learn. So while we are setting up the table, we have seen whether it was COVID, whether it was HIV, the data which was shown to us, that the gut immune system is the main immune system for both as we are growing and in the adult life. And the infections like COVID or HIV, they destroy the gut immune system completely. And the beneficial bacteria, if they encounter the immune system for the first time, they have many methods by which they build it up. They do it through receptor mechanism, the toll receptor mechanism, the pattern-seeking receptors. They have a huge number of systems to really build the gut immune system, and then gut immune system goes everywhere in the body because there is a homing pattern which happens. So the whole body benefits out of that. There are a large number of cytokine, exocrine, and other things which are secreted by immune system. So we know, as far as probiotics are concerned, that they bring down the inflammation, they build up the immunity. They also act on the autoimmune system, bringing down allergies and many other things. And sometimes they are beneficial in some debilitating diseases like cancers and others. Many times, one of the things which we don't know whether they can attack the cells. Yesterday, there were some very good papers when in the symposium here, which I attend, I saw that some of the systems in the HBV infected cells, they might have attacked the cells and harmed the liver also. So the probiotic, when they populate, it becomes good. The diversity is also very, very important in this, because more diverse population it is, the better clonal expansion offers. So with these comments, I will invite my panel, which is a very elite panel, the Dr. Amulya Panda, who is the former director of National Institute of Immunology and is a fermentation specialist, as well as has a deep knowledge of immunology and vaccinations. Then we have Dr. Pramod Garg. He is the director of THSTI. He comes from the All India Institute stable of the famous gastroenterologists and understands at this given moment, THSTI has one of the strongest microbiome or gut microbiota or metagenomic group over there. And they have really benefited by the presence of Dr. Pramod Garg. And as you saw that the population expansion which occurs, the FMT, which was the symposium which we had yesterday, was very, very important for the ultimate outcomes. So even the constipation is very, very important thing in this population. Then we have got N.K. Mehra. I call him Dusht Balak because he hasn't got He has a mischievous trait and hasn't got a single gray hair in his head. He is one of the first. He established the. There was in the All India shoot. There were 2 or 3 immunology points. One was in the biochemistry department, spawned by Dr. Talwar. Another one was Dr. Malviya, the clinical immunology. But he established the genetics, immunogenetics, and histocompatibility group there, which did some very good basic immunology. And I consider him as a strong immunology person. And he has been the president of the Immunology Society also of India. Then we have Dr. S.K. Mittal. Dr. S.K. Mittal is again, he did his graduation from All India Institute, the famous pediatric clone, and later on was the head of the Maulana Azad Medical College, and has been in our governing body and is a very competent pediatrician. We have Dr. Kea Lahiri. She is also a pediatrician, and she actually has a lot of experience in vaccinations and has really very hard data on these areas. So we will ask her some questions on this particular area also. Then we have Dr. Nirupama Trehanpati. She is a professor and head of the molecular medicine department, which is essentially a research department of the ILBS. And Nirupama is a very good, competent immunologist, and she has some very good matrices to really measure the immune responses. And when there was a symposium over here, her matrices and immune responses were studied, that how the gut microbiota could be used, or the dysbiosis is related to the HPV infection. But Nirupama told me that your questions were not good, so I have my own questions, and I will take. So Nirupama, you will have opportunity to do that also. So my first panelist is Dr. Amul. I'm very grateful for all the panelists to be here. So we have a full house and full quorum. And I will start with Dr. Amulya Panda. One of the major things, Dr. Panda, in the probiotic that we don't understand, because I worked in the fermentation in the vaccine field. It was very difficult to get even 10 to the power 8 or 10 to the power 11 bacteria growing in the fermentation, because even if you replenish the media, it didn't happen. And so here we have to give billions of bacteria growing in the fermenters, and then later on distributing it in the vehicle. So how does it happen in the PEV? What we need to do that we will get an appropriate numbers? This is my first question. Yes, please.
Thank you, sir. Yeah, I am like an odd man out. I am at the end of probiotic in the production chain, and as I hear, everybody wants probiotic. Imagine everybody wants to consume. You need a huge amount of sachet. And please remember, growing them in the fermenter is a real challenge, because these lactobacillus, bifidobacterium are grown. E. coli is growing. Many people are growing. The maximum safe you get is 10 to the power 9. That is 1 dose per ml. But if you want to go beyond, because you want to meet the demand, you need to understand how these microbiome, the organism grow. In fact, if you see, most of them produce lactic acid, huge number of lactic acid. In fact, if you grow lactobacillus or bifidobacterium, the concentration of lactobacillus in the fermenter will go 30-40 gram per liter, which will be inhibitory. So the real challenge to grow from 8 dose, 1 billion to 5 billion, 10 billion, in fact, the new technology has come, something like mammalian perfusion culture, where you can remove the lactic acid. there are different fermentation going on. In fact, latest news is that you can have 10 billion cells per ml. In fact, that is tenfold increase in the amount of CFU by going this perfusion culture. Or you can take out the broth, remove the lactic acid and start fermenting. This is one. The other thing is coming as we keep on discovering new organisms like lactobacillus, even propionobacterium, even Saccharomyces. There are certain organisms who are probiotics, but they grow on lactic acid. So the ideal combination will be, can you have a mixed culture fermentation where you can grow both lactobacillus, a propionobacterium, or a yeast who can consume the products lactic acid, and so that ultimate growth can be done. So technology is available. In fact, the way demand is coming, it will be almost important to develop this new fermentation technology. And remember, all of them are all anaerobics, most of them. So the growth is very, very limited, unlike aerobic culture. But with new improvements in recycling, extractive fermentation, doing mixed culture, it will be possible to get at least 10 billion CFU per ml of culture, and that will significantly improve the production cost.
Thank you for clarifying this. So one of the tricks is creating a new fermentation and modern fermentation technologies and removing the lactic acid, either by creating a partner or alone. When I come to the next part, I gather that formulation is very, very important. And in this formulation, what will actually, one of the things like in the LCS, we provide two properties, that we make it acid resistant, we make it bile resistant, so that it can survive within the gut. But what were the tricks in formulation? These days, we add prebiotics, we add symbiotics, we add so many things to make them work better. So what are the tricks in the formulation?
In fact, the real challenge in producing huge amount of probiotics, one is you grow them, nice fermenter, but the second and third is you purify or make formulation to make a product. But more important thing is you store them so that they become alive, viable, because at the end of the day, we are delivering a viable microorganism to a place where it should reach, to the colon, by passing through stomach and other things. So the formulation is important on two aspects. Number one, it should protect the organism, it should make it live, its potency, and more importantly, it should go into the gut, passing through the stomach. What people are doing nowadays is, keep on adding prebiotics, like many polysaccharide. It can be plant polysaccharide, it can be microbial polysaccharide, it can be seaweeds, so that these polysaccharide, they protect, they get hydrolyzed and become prebiotics for the formulation. The other most important thing, which many people may not be knowing, is the most important thing for the probiotic manufacturing is the water content and vapor phase transformation. Because how do you lyophilize and store? Because at the end of it, the lyophilized powder has to be dispersed in a liquid, and on its liquid, it should be again viable. This is important for storage and stability, and you need to take care of the water content, because water activity is more important for maintaining the viability. I think nowadays what people are doing is they are adding prebiotics along with that. In fact, a lot of encapsulation technology is also coming up for the formulation. They are entrapping these probiotics in alginate, in polymers, in carbohydrates, and many things. In fact, many technologies are available nowadays where you can entrap or encapsulate spores in biodegradable polymers or prebiotics and can be used, so that it protects, it maintains the viability, as well as it helps in both pre and probiotic mixture.
So that's a very good thing. These capsulated entities could perhaps be also used as an alternate to FMT, the fecal microbial transplantation.
Yeah.
So the main trick, as you say, is to make them viable and provide them compressed carbohydrates and other things, so that they could ultimately pass the barrier of the stomach and they survive. So survival is the major thing.
It's not only survival, sir. It's you have a probiotic powder. At the end of it, you need to dissolve it to eat it at certain day. See, if it doesn't reconstitute again and maintain the same viability, that became a real challenge.
But do you think that milk will be a very good carrier?
In fact, people have been using milk and whey protein to make powder during spray drying. In fact, it's already on.
So thank you, Dr. Panda. Now I come to Dr. Pramod Garg and give him a very difficult question. We were actually one of the gastroenterologists in Gangaram says that dysbiosis is now a very major disease entity. Could we really check who has dysbiosis, and could we treat them for with the appropriate microbiome restoration or replacement, so that they get rid of the dysbiosis? And could we charge these patients so that, because this will be expensive to check the restoration whether it has happened or not, and diagnose that it is dysbiosis with the laboratory test, not the clinical test system. So my question to you, Dr. Garg, is this: that how do we deal with the dysbiosis in practical life? And do you think it is a distinct disease entity?
Thank you, sir, for this question. Can I ask in the audience, how many of you are microbiologists or nutritionists or physicians? Physicians, if physicians, there are a few. Nutritionists, okay. Dietitians and nutritionists, same thing. And microbiologists? Okay, thank you. So there's a mixed crowd. So first of all, regarding the term dysbiosis, dysbiosis simply means that eubiosis is not present. The problem is we don't know what is eubiosis, and therefore we don't know what is dysbiosis. And the reason for that is that every individual's gut microbiome differs from the other one. There are certain broad categories where you can say this is likely to be normal or this is likely to be normal. And there are so many differences between gut microbiome, different groups. If you take urban and rural, Indians and Japanese, and this year from PHSTI, who has shown that Denmark and the Indians' gut microbiome are also different. So the influences on our gut are immense because of diet, because of antibiotics, because of proton pump inhibitors or acid suppressants, because of natural GI infections. So therefore, to define a normal gut microbiome is very difficult. Having said that, for an individual, a gut microbiome is from early childhood onwards stable, unless there are problems in between. For example, a GI infection, gastroenteritis will lead to some perturbations. And then you call it dysbiosis. So someone is doing well, gets an acute gastroenteritis, and I think most of us have had that. Either it's a food poisoning or acute GI infection. And because of acute GI infection, afterwards, when the infection has been controlled, you find the person is having some GI symptoms, some abdominal pain, some bloating, some nausea, some vomiting. And we think that now is a state of dysbiosis because the normal gut microbiome has been altered by that infection. And in this situation, if we treat with something, then we expect that microbiome to come back to normal homeostasis, eubiosis state. So to that extent, dysbiosis is okay. But otherwise, in general, to define dysbiosis may be difficult. What the previous lecture also showed, that if you take groups of patients, whether it is diabetes, Parkinson's disease, any other neurological disease, fatty liver disease, another disease, coronary artery disease, and you take a healthy person without any over disease, you may find differences in the gut microbiome, so-called signatures between the two groups. And then it becomes interesting to know whether these changes are related causally to the disease, not just association. So I think this is a very, very important question, very difficult to answer. But in general, short courses of dysbiosis, short duration of dysbiosis is well known following infections and antibiotics. Long-term dysbiosis is more difficult to define, but has been shown to be associated with disease groups.
So IBD is not dysbiosis?
So IBD again is a very complex disease. Now to say it is just dysbiosis is not correct. So there are three major, if not causal, associated, significantly strongly associated factors. One is that there is some kind of a genetic predisposition to that. Second is that there is possibly altered gut microbiome. What preceded, we don't know. Whether the gut microbiome got altered after the disease or before the disease, that's the question. And third is that there is immune dysregulation, not hyperimmune, but dysregulation. And that's why it's come something like an autoimmune disease. So gut microbiome does play a role. And that is the reason why in ulcerative colitis, and by the way, IBD is of majorly two types, ulcerative colitis and the other is Crohn's disease. In ulcerative colitis, there is some evidence that probiotics might help, not so much in Crohn's, but FMT or fecal macrobiotic transplantation does help ulcerative colitis.
So ulcerative colitis, the one of the pathogenetic factor in ulcerative colitis is the own immune system turning into autoimmune system. And so some of the therapies are also the same kind of like Humira, etc., which are used in the autoimmune diseases. So how does a probiotic, the only evidence we observed which passed muster was the pouchitis. But what is the other situation, that how does it work in the and why it doesn't work on Crohn's?
So I don't know that simple answer. But what is believed is that if there is altered gut microbiota or dysbiosis, and there is underlying genetic predisposition, which puts your immune system at risk, some of these bacterial or some other microbes may dysregulate the immune response of the body, which is generally to maintain gut homeostasis. Now, if you replace that gut microbiome with a so-called healthy microbiome, now this will not stimulate your immune system to that extent or in a dysregulated fashion. And that is why fecal macrobiotic transplantation has been shown to be helpful.
So do we give it during the remission system period, or do we give it the 4-week period?
So there are two periods. One is the induction of remission. That means someone has active disease, having symptoms, and then you give a treatment to induce remission. And then you give treatment to maintain that remission. There are two important things. So FMT has been shown to both induce remission and also maintain remission. And similarly, there is some evidence that probiotics may also help induce remission and also help maintain remission.
So in the why it should not work on the has anybody worked on celiac disease?
There are some reports, but I don't think we can say that probiotics is a treatment for celiac disease. Celiac disease in more than 95-99% is a very simple disease to diagnose and treat. Just stop meat and you are okay. I don't think we should even recommend probiotics for celiac disease.
Actually, yesterday we had an FMT workshop, and exactly what you have said, that there were a lot of discussions on who is the normal. And, and that discussion was very interesting. People have different definition, but I agree with your definition of normal, because that is the best we could get at this given moment. So thank you very much, Dr. for this. One more question, because yesterday that question came up, that should the FMT be given by nasal duodenal route or through colon route?
So you are all asking all the difficult questions. So, I mean, basically, we want colon to be populated with healthy bacteria. That is the major goal. But you should understand that even in the distal small intestine, there are lots and lots of these microbes. So if it populates distal small intestine also and also colon, that is better. For ulcerative colitis, at least, or for C. difficile infection, largely introduction into the colon is effective, and that has to be done through colonoscopy. That means we do endoscopy and deliver it. The other way is to put a tube into the jejunum through the nasojejunal route and rice tube and give it through that. Both have been shown to be okay, but both are associated with some patient discomfort, and also it needs expertise. The upcoming therapy, which is likely to come up, I think, in the next 3-4 years, is that you have lyophilized fecal samples like capsules, like probiotics, and then someone can take it orally. I think that would be the best. The only question is whether they'll be as effective as the natural stool or not. That I think we need to see.
One more question I would like to ask you, that whether in your journey, have you ever thought, because there is a specialized area, to use the microbiome pathway for acute pancreatitis?
So there were actually a study, randomized trial about 2006, 7 or 8, as a Dutch multicenter trial where they did give probiotics to patients with acute pancreatitis. And let me give a background to that. In patients with acute pancreatitis, in the second or third week of illness, they get infection, secondary infection. So acute pancreatitis is a condition where you have acute inflammation of the pancreas and some damage to pancreas. And this leads to necrosis and some fluid collection inside the tummy, inside the abdomen, which may secondarily get infected, which is called infected necrosis. This occurs in second, third, fourth week of illness. And it's a fairly serious problem. So what the idea was that these infections probably arise from the gut. So if you give them probiotics, you might help these patients prevent the infection or decrease the morbidity and mortality. Unfortunately, the trial proved futile. Rather, more patients in the probiotics group died, and the trial was stopped prematurely. And even though it was a negative trial, it in fact showed harm and stopped prematurely, it was published in Lancet, just to make sure, prove the point that not everything is hunky-dory.
Thank you, Dr. Garg. It has been absolutely fantastic. Most difficult questions were thrown at you, and you did very well to clarify them. Dr. Mehra, I have again some very difficult questions to you. Firstly, that you know that as we grow, the thymus becomes an important thing. There are so many areas from which our immune system is populated and matures. So why do we call gut immune system as one of the major immune system? What is the rationale behind it? And I will also like to understand that what is the mechanism of these strains, which are so-called probiotic strain or so-called beneficial strain, to really build up the gut immune system and again turn them towards beneficial populations or harmful population? How does it happen?
Thank you very much. I think your question has two components. One is to talk about the gut-associated lymphoid tissue. Now, gut immune system provides a physical barrier. And this barrier is the epithelium, and below the epithelium, as you know, is the lamina propria, which is contains the immune cells. This constitutes what we call the gut-associated lymphoid tissue, GALT. GALT is a part of what we call MALT, is a mucosa-associated lymphoid tissue. And GALT, of course, constitutes the largest component of the immune system in the humans. And GALT is considered as the largest immunological organ, as it contains approximately 70 to 80% of IgA forming cells. The IgA is the most important antibody over there. these antibodies are locally synthesized and provide protection on the mucosal surfaces by binding to and neutralizing the foreign antigens that come through the food. By the way, the recent evidence also indicates too much of IgA antibody is also not good, because the too much of IgAs could have also act as inflammation players, both at the mucosal and the non-mucosal surfaces. But immune cells, and your second point was on the probiotics or the gut microbial flora, as we call. So immune cells and gut commensal microbes in the human intestine constantly communicate with each other so as to maintain healthy immune status. The immune system microbiota crosstalk relies on a complex network of pathways that sustain the balance between immune tolerance and what we call immunogenicity. This is also true of the probiotic bacteria, which can interact and stimulate intestinal immune cells and commensurate and sort of commensal microflora to modulate the specific immune functions and immune homeostasis. I may also state that only a few studies have been done so far, demonstrating a beneficial immune modulatory effect of the probiotics. But some of the studies that have been done, they consumed probiotics specifically mediate the activation and modulation of both innate immunity as well as the adaptive immunity. And this is done by two ways. One, production of various cytokines, chemokines from the dendritic cells that are there in the intestine, lymphocytes, macrophages, mast cells, granulocytes, and the very important intestinal epithelial cells. And second, as I said, about the IgA forming cells. Probiotics can improve the host immune system and induce important beneficial effects, allowing the prevention and or management of immune or inflammation related to the gut, including the IBD or inflammation, diarrhea, pathogenic infections, infant colic, and certain cancer types. But I think the last word is that the important question still remains as to how probiotic the bacteria and immune cells interact to improve the immune functions. It has only been shown for the respiratory infections. In fact, I saw a study that people who take the probiotics can really resist the COVID-19 infection as well. But COVID-19 has also been shown to be to those who take the regularly there. So I think there is some evidence in terms of improving the respiratory tract infections, but I think more studies need to be done to understand the interaction between probiotics and the immune cells in the gut.
Why some children who are given cow's milk or buffalo milk develop the allergic reaction to cow's milk or buffalo milk protein?
Yeah, that is also true. I mean, that kind of a thing would be definitely true. I think there the genetic factors, the familial issues, those will be very, very important there. But not everybody will develop that the reaction to cow milk or
Do you know any study where the microbiota is done somewhere? Dr. Mitchell and Dr. Kaya, you could also answer when the time comes.
Cow's milk. Now, it's not in relation to the cow's milk.
Modifying allergy, giving probiotics to the mother and to the infants. Those studies have been done, though not with much benefit.
So may I add?
Yes, yeah.
So there's a hygiene hypothesis, which is very important for the TH1, TH2 balance. And when that gets disrupted, you have children who are more prone to allergies. Because if the child is born in the farm, they have seen that they are less prone to allergies. And respiratory infections, allergies, especially allergic rhinitis, has been found to be very effective because we have done a small pilot study at DY Patil Hospital where we have found the PFT increasing in a better way, though we cannot really say because it was a very small pilot study. But I think there are studies where they have reported allergic rhinitis responding to not only probiotics, but vitamin D as well.
So, Dr. Mittal, I had approved a study of giving probiotic Lactobacillus plantarum in infants in Odisha for a septicemia study. So a group of people had filed a case against me that you have endangered these infants. Although later on 25% benefit was seen, and later on ICMR did a study under Vinod Paul and others, and the same thing. But there is a perception among the judges and among the general population that if you give infants the probiotic, it may harm them. What do you think?
Sir, till now, all these studies which have been done with probiotics administration in children at different stages.
Now, my thing was infant, not children.
Yeah, I'm talking of infants also, sir. Even in infants, in all stages of their life, that is in the antenatal period, to the mother, to the preterm babies, to the neonates, to the infants as such, at all age groups, the probiotics which have been largely used, that is the lactobacillus, the bifidobacter, and streptococcus thermophilus. These have largely been, in fact, hardly any side effect have been reported. Only one, enterococcus, we are not very sure. Although even that has been tried with certain infant milk preparation, but whether that may prove to be providing some sepsis or something, we cannot say at this stage. But otherwise, the lactobacillus, bifidobacter, and they have been tried in almost every thing. In premature, to prevent necrotizing enterocolitis, to treat neonatal sepsis, in infants to treat rotavirus diarrhea, to treat influenza virus, to prevent respiratory infections in the perinatal period, to decrease the episodes or allergy episodes in allergic diabetes children.
So suppose accidentally I end up giving probiotic to a child with dysglobinemia because they get a lot of infections repeated, but it really doesn't come out for a pretty long time and they remain undiagnosed. So if we give them who have primary immune deficiency, there are several other conditions accidentally, what will be the danger?
Sir, in primary immune deficiency, it is difficult to say because they will have a combined both cellular mediated immunity and the immunological, that is the cell mediated and the humoral immunity, both are deficient in the combined immunodeficiency. In these, even the so-called harmless microbiota like these probiotics may become this thing. But these conditions are so rare, so that I do not think that they should really be coming into question in the largely the normal children that we are talking about, the safety of probiotics, because the incidence of these combined immunodeficiencies and all extremely rare.
So the risk-benefit ratio is very
not
very
enough
small.
numbers to say whether they are safe in that or not. It's very difficult.
So, Dr. Kia, since you are very adept in doing vaccine studies and have a theory for the probiotics will improve vaccine response. The only study which I read, which was very, very critical, was Parallel Ogra's study from the Buffalo, where he showed that polio responses in children are better if they are given probiotics. Very old study. He actually presented it in India when he came here. Are there really solid evidences that if we give probiotics, there are all theories with vitamin A and response. Are there solid studies that if we give probiotic, particularly when we go for a mass vaccination, there are children who are preterm, who are extreme preterm, who are low birth weight. They are essentially immunologically challenged. Are there any real solid hard evidence that probiotics will enhance vaccine response?
So I think I'll take your preterm question first because that's a very important point. At our hospital, we are giving preterm babies rescue need, that is Bifidobacter breve, which is 0.5 grams sachets, which is given diluted with breast milk. We can dilute as well, and distilled water. And it's given once a week. Now it's given to prevent NEC and sepsis, which are common, and to increase the enteral feeds to oral feeds and reduce the hospital stay. Now, I'm not talking about less than 1,000 grams and less than 27 Bs because they're extremely preterms. Now, one must remember that preterm babies are not IUGRs. They have not retarded or they have suffered in utero. Once they're given a good environment outside the uterus, they challenge the term infants and they do as well as the term infants. Now, we should remember that we have to immunize our children. In US itself, 50% of preterms are not immunized because they have a longer hospital stay and they go away unimmunized. In India, it's not such a bad situation, but post-COVID, things have gone haywire. It is very scary. We are trading one disease for another. We have a measles outbreak. We have a H3N2 outbreak. Thank heavens if polio doesn't come back because we have eradicated polio. So the point is that why not add an adjuvant? That's a probiotic, because that would enhance seroconversion and improve the protective effect and give them a bifidobacter in a nice way where they can be protected. It is worthwhile giving it because it is safe. And by grass also it has been proven safe. And there's a very good article in 2022 by Fortman et al. Where they have shown the efficacy of probiotics in preterm babies.
Thank you. One more question I will like to ask you. That those vaccines which go with a lot of adverse reactions, like the COVID vaccines, they had a lot of adverse reactions. Will giving probiotic reduce those adverse reactions?
So I wouldn't play around with a new virus like COVID and children because we should give them the basic immunization first. And we can start at any time. I think everybody in the audience will deal with immunization in some way or the other. You don't have to be a pediatrician or any other specialty because if you do not immunize them now, you will have diphtheria, pertussis, tetanus, hepatitis, name it, and you have it. Influenza having very different variants. So we are not sure what we are getting as vaccine. And it's very, very difficult to say that we are really adequately covering up influenza. So the point is the basic immunization. If they are below 5 years, give them at a 1 to 2 month interval. There's a special schedule. More than 5 years, the DT is given because children have missed their vaccinations. And that is the first primary event. We could add on even for mass screenings, we could give except for immunocompromised and critically ill children because there can be translocations, which have been proven. And these are the only two contraindications. But preterm babies have either cesarean sections, they do not have the adequate breast milk, they do not have the skin-to-skin contact, and they are IgA low. So the point is now that probiotics would give a definite immune modulation to these patients.
So our last panelist is Nirupama, and most important panelist. Nirupama, Japanese scientists did a study in which they found that those who were chronic tobacco users, if you give them probiotic, the immune responses improve. And that will be beneficial. That was there. When we did that in India, we didn't find the same because there's variation in population. And tobacco use is very, very difficult to really measure. So we didn't find that particular result. So although it was monitored by the same group who had done the earlier study. So one thing becomes very important, that what is the matrix of responses which we should measure, that these responses, if they are there, they will be overall protective, whether it is cancer, whether it is infection, whatever it is. So I saw your data. In that, it was shown that that CD8 cells with activation markers, with later on enzyme came up also, is one of the markers. In the same way, the NK cell, one of the NK cell population out of the four, or the plasmacytoid dendritic cells, or the dealing with the checkpoint inhibitors by removing those things, even in the infection, when you did a hepatitis B infection, will be one matrix to measure them. What is the fidelity of these matrices that it works in some people's hand, it doesn't work in other people's hand? So how do we create a very solid matrix of measurement of an immune response that the baseline was this, and how many fold should it increase? Twofold, fourfold to make it significant.
Thank you, sir, for giving me the opportunity to be here. But your questions are many, so I will go one by one. So your first question was that the tobacco study which you conducted and there was not response. So question is,
And NCS guys conducted it in Pune.
I'm not aware of that study, but which panels and what
It was
immune
not published
response?
because we didn't get it.
Yeah. So the first question is because when we give the probiotics, it really gives the more of the IL-6 and the interferon. And IL-6 and interferon does not only secreted by the TH panel, and that it is basically NK cells and basically the dendritic cells, basically the antigen presenting cells which are capturing the probiotic in the gut and then giving the response IL-6, and even in fact the probiotic broken material is gone to the lymph node, and which gives the better cells in the even the B cells differentiation also. So I believe that we should start with the dendritic cells or any antigen presenting cells and then move on to the study for to see the T cells or the B cells. But we are missing here the one point. Whenever there is a kind of a probiotic we are giving, or we are seeing that differentiation, so we are giving even the vaccine and the probiotic, there should be the T follicular helper cells which should be seen. Now your second question, that the matrix is not working in the one person, but it is working in the other hands. These are the really fixed matrices. Only the difference is that what kind of the combination of the flow markers you are picking up. So we know that the lymphocytes or the CD3 T cells are abundant in the all leukocytes. So we can certainly pick the very weak flow marker, and the regulatory T cells CD25 are the very few cells, few events. We should pick up the very strong marker, flow marker with that. So these combinations certainly, because the flow panels are always, it should work in each lab. And if it is, I have standardized, and if you go to the Gangaram or the Artemis, it should work. So it's not like this, that it won't work.
So actually,
and it's depend upon that
the olden days, myself, Dr. Indianath, and several others tried to see that what is the fidelity of these measurements. So the equipments were different, the reagents. So we standardized the equipments, standardized the reagents and others. We got fairly better results with the time to time. So and the expertise is not same with everyone. So you said that when the cell numbers are small, the errors creep in.
So, sir, if we know that the panels that the regulatory T cells are very rare events, or you can say that I go for the B cell panel for the 16 color. So let's say that I have plasma cytoid cells. It is very rare events. So for the rare events, you use the better markers, and then you can pick up very early. And then you have to use
twofold
the
increase or fourfold increase?
it will be I think the 2 to 4-fold increase when you give the probiotic or the vaccine response. It is less than if it is twofold, it is not, it does not make any sense.
So the fourfold increase will be definitive.
Sir, you can't say the 2 it's a standard deviation in the population
So how
much.
do we know, this is the last question, how do we know that whether a person is a responder with the probiotic or a non-responder? Yesterday in this, there was a group who was responding, who was not responding.
Certainly, we should see with the probiotics, fecal cytokines. Sir, it's not the immune cells. If you measure the fecal cytokines, it will give you the real results. Now we are on to that fecal cytokines.
Good.
Yes.
Very good. So I thank the panel profusely. We would have loved to have questions from the audience, but it is the end of the day. Audience is thinning up. But tomorrow and during the lunchtime and the other times, you are invited to clarify anything which you didn't go. But I will allow 1 or 2 hands if they go up. Yes, please.
Very good evening. I'm Dr. Sushant Vadera. I'm a surgeon, laparoscopic and bariatric surgeon. Dr. Gangli, I was previously in Gangaram. So yeah, it's good to see everybody here. So I had some questions to various panelists, starting with Dr. Panda here, sir. As you said, you have difficulty in growing lactobacillus because you cannot meet the demand. There was a study done in the Swedish population. They used healthy vaginal secretions of healthy women without any candidosis or any bacterial vaginosis. And they were able to isolate lactobacillus, mostly the acidophilus strains. So can be used, because that's a substrate, or that can be a very good fermenting area to grow lactobacillus, but a different species, not just the acidophilus. And that might actually have a solution to your problem, because that is a very rapid growing and a medium for lactobacillus.
No doubt that is exciting, but please remember the volume is huge. You need billions of liters.
You would not need a healthy woman's vagina would be.
No, that organism is okay. What I was talking is, if we want to supply to many people across the world, you need to grow that. That milieu may be good to isolate an organism, but to replicate that, any lactobacillus will end up producing, because that is the inherent quality of any lactobacillus. They consume glucose and they will produce lactic acid.
If you give the mic to Dr. Talwar, the question is this, that the vaginal secretion might help growing the beneficial lactobacillus better.
Well, I think it's worth trying anyway, but there is no answer on this issue to my knowledge. But it is worth trying.
Actually, Dr. Talwar's work showed that some of the special population of lactobacillus grew better in the female reproductive tract. But as Dr. Panda said, that if you are making an industrial product, that is not a practical way of doing unless we identify the protein or any other thing which is growth benefit.
No, what I can add is the isolates from vaginal tract may be more resistant to acid environment. So there, the chances of toxic growth by lactic acid will be low, so that probably you can grow more.
Yeah. Yeah, that is what he's saying. That many times the lactobacillus growth is hampered because of the lactic acid secretion. So vaginal fluid is naturally low pH. So he says that that might be one of the reasons. So I think this is the question which we had. Yes, please. Very brief question. And identify the panelists with whom you are asking.
Hello, sir.
Yes.
Yes. So my question to Professor Nehra, you know that the probiotic we are eating like a magic bullet. So we always feel that it will work in neonates, it will work for children, it will work on the healthy population, it will work for the inflammation, respiratory, everything. Do you think that is true, or do we need some disease-specific probiotic that can give better output instead of using only one probiotic?
Who is the panelist whom you are asking?
Professor Nehra.
Mehra.
The best immunologist I know.
The question is towards you.
बीच में आवाज कट रही थी, मुझे सवाल की समझ नहीं आई.
So, sir, what I am asking that
थोड़ा सा जोर से बोल के माइक को पास में लेके.
Can you hear me? Okay, yeah. So what I'm asking, instead of using probiotic as a magic bullet, whether we can use disease-specific probiotic, because the same microbes may not be ideal or optimal for each and every disease. So what is your comment on that?
He is asking, Dr. Mehra, he is asking that each microbe might have a particular signature. Are there microbes which are better for building immune responses or building and one type of immune responses to the other? Because he carried out a study in which he did Lactobacillus rhamnosus and find out 23 signatures in the Lactobacillus rhamnosus. So
And you can also look into the oral probiotics. You know, you take oral probiotics every day, yogurt and all that. And what this oral probiotics do, and that might answer your question also, they interact with the IECs, the intestinal epithelial cells, or the immune cells, and in the lamina propria through the toll-like receptors, the TLR. So I think TLR study, TLR polymorphism is also known to influence and all those kind of things can be looked into, because these then induce the production of cytokines, chemokines, and one of the other proteins that we know is very important is what we call MCP1, the macrophage chemoattractant protein 1, which is also secreted by the IEC, the intestinal epithelial cells. And they send signals to other immune cells, leading to the activation of the mucosal immune system there. Now, whether or not this system, the oral bacteria, the oral probiotics are equivalent to the probiotics that you give is not yet fully aware, but we know that the mechanism, one of the mechanism is that they activate the regulatory T cells. And these T cells create the interleukin 10.
So one of the answers, if I can understand Dr. Mehta, is also that pattern seeking moieties are very, very important. And some of them do particular toll receptor, like anything which affects the toll receptor 9 may move it towards autoimmune disease. Anything which does toll receptor 2 will go with the immunity and vitamin D and other responses. Anything which will interact with toll receptor 4 will move it towards inflammation. So this is the best thing, but you perhaps if you have the answer, you can give the answer. Otherwise, we move one last question. Yes, later. There is the last question today.
Hello, sir. My question is, could we think about the postbiotic in terms of bacterial metabolites in place of probiotic? So we can also deal with the treatment of the patient with heavy like HIV load, as we have previously discussed. And we could also deal with
the different status of the patient with the different bacterial strain. And if we could think about it, what will be the major effect?
Whom you are asking this question, I'm not very clear about the question. Could you phrase the question?
You only. He's asking you.
I'm not a panelist, but if I understand your question, you said that because one of the things which came up was the metabolites,
Yes.
which the probiotics affect,
Yes.
and the last thing in the Parkinson's disease you saw that one of the major method in which these probiotics were acting, that they were producing metabolites which could pass the blood-brain barrier. And the metabolomes are one like the butyrate, lactobacillus they produce butyric acid and propionic acid. These are very good for diabetes, these are very good for obesity. And these metabolites, there are metabolites which could be replenished. So one of the things which this institute does, that Jaswinder Singh from this institute runs the Orbitrap LCMSMS, and they do the metabolomes. And these metabolome signatures are very good. Many times, tomorrow you will hear an article or a lecture where you will find that how these metabolomes are linked to a particular strains of the probiotic strains. So there is this is the science for today and tomorrow. And your question was very good. And this perhaps will later on will bring in designer entity which will be able to deal with it. Thank you very much. Thank you.
Thank you.
All the audience to be fair and all the panelists to be so patient and answering so nicely all the questions. Thank you.
That was quite a remarkable panel discussion indeed. Can we have a round of applause for the moderator as well as the panelists for curating the entire session diligently? And I would now request Professor N.K. Ganguly, sir, to kindly do the honor of presenting mementos to our panelists here with. Can we please have them? Starting with our panelist, Dr. Amulya K. Panda. So please step forward. Yeah, thank you. So please accept this token of gratitude, and thank you so much for your humble presence here today. Everybody, can we have a round of applause? I know it's the end of the day, but I would request you to, the louder you clap, that will be more energizing to our delegates here. Okay, so we have Dr. Bhavtosh here with us. Okay, thank you so much. Next, I would request Professor N.K. Mehra, sir, to please step forward. Yes, sir, please come. I would again request Professor Ganguly to do the honors. Everybody, can we have a round of applause, please? Thank you, thank you. Next, I would like to call upon Professor S.K. Mittal. Professor S.K. Mittal, sir, please take center stage and accept this token of gratitude from our end. Thank you. Next, I would like to call upon K.R. Lahiri ji to please come forward. Ma'am, thank you so much for your humble presence here today. Please accept this token of appreciation from our end. Last but not the least, Dr. Nirupama Trihanpati ji to please step forward and accept this token of appreciation from our end. I would also request the humble presence of Mr. Iwama, sir, to please come forward and, of course, felicitate the moderator of the session, who has very diligently conducted the entire thing, Professor N.K. Ganguly, sir, here. Yes, sir. Sir, it's on the way. We are just waiting for Mr. Iwama, sir, to please come forward. Thank you, sir. Please do come. and present this token of appreciation to the moderator, Professor N.K. Ganguly, sir, here. Thank you so much. Last but not the least, I would also request the humble presence of E.G. Amano, sir, to please come forward. And I would also request the humble presence of Dr. G.P. Talwar, sir, to come and accept the token of appreciation. We have Mr. E.G. Amano here with us, and I would request sir to please wait half a second. Please step here. So we have the photo op happening with the entire panel crew. I would request Dr. Talwar to please take center stage. And I would now humbly ask Mr. E.G. Amano, sir, to please present this token of appreciation to Dr. Talwar here. Everybody, can we please have a round of applause? Louder, come on. Hands up in the air. Dr. Talwar, please accept this token of gratitude. And thank you so much for gracing us with your humble presence here today. I would request all the dignitaries to kindly pose for a photo op now. Thank you all. Thank you, sir. Thank you. I have a few special announcements for all of you, so kindly stay put. The certificates will be available after end of day 2 of Symposium. Tomorrow, our sessions will begin at 10 a.m. So I request you all to kindly be present here by 9:30 a.m. All the delegates who would be logging in online, you have to join us sharp at 10 a.m. Also, don't forget to fill the feedback forms online and submit it online. That will be all. As all good things come to an end, today's event, day 1 of the symposium, is also coming to its closure. It was wonderful to have you here with us. See you tomorrow positively, sharp at 9:30 a.m. This is Anchor Chandni Sehgal signing off for the day. Thank you so much.